The dragonfly is extraordinary, and its flight is like a dance. It can fly forward, backward, upside down, or make extremely tight turns. But don't be fooled by its beauty. To feed, she is merciless. It is a very good hunter. She is a flying ace perfectly equipped to survive in her environment. If she is as greedy as her offspring, the other insects have something to worry about. If we could zoom in on certain parts of her body, you would see that her eyes give her a 360-degree view, that her legs are equipped with small hooks, and that her jaw and mandibles are very powerful. It can see all around it, grab its preys, cut them up, shred them, and swallow them. There is another detail that you should also observe in this creature. Look at its abdomen and flanks. You see these little holes that seem to open and reform, a bit like trapdoors. These are spiracles. These spiracles pump air into the dragonfly's body through its respiratory system. It is very different from our pulmonary system, but it is efficient. Still, to make powerful muscles work, a lot of oxygen is needed, and the spiracle system is not nearly enough. This is probably why dragonflies today are 6 to 7 times smaller than our giant dragonfly species. You may wonder why this insect was so big. You'll soon find out. It's far from being the only one. One of the explanations put forward by scientists lies in the oxygen content. The oxygen level in the Carboniferous period was higher. Today it is 21%, but at that time it was about 35%. This means that for every breath the dragonfly's body receives much more oxygen than it would today. This assumption would explain why insects were so much bigger, but also how such a heavy insect could manage to fly. Flight requires strong muscles and a lot of energy. Oxygen is one of the essential keys to flight. However, as we saw earlier, spiracles have their limits. Only a much higher oxygen content than nowadays could explain the feat the giant dragonfly was capable of. Let us leave our here to its hunting prey to take again the road. We have an appointment with a millipede, and not just anyone. If I tell you millipede a few minutes ago, you would have probably imagined a small beast of about 10 centimeters. But now that you've met the giant dragonfly, you'll know that it is definitely bigger than that. Arthropleura was a kind of arthropod, a distant cousin of millipedes, which lived in the Carboniferous period in a continent that no longer exists today, Laurasia. It was as long as a city car and was almost 3 meters or 10 feet long. Its name means articulated ribs in Greek and illustrates its flattened body, composed of about 30 articulated segments. Each of these segments is covered by 2 side plates and a central plate. If it is long, it is also very wide, about 50 centimeters or 20 inches for the biggest. In terms of proportions, it could have competed with the alligator. Can you believe it? Never before watching this video would you have thought that a millipede could be compared to an alligator. It would have seemed completely unbelievable to you. Well, now you know that in the Carboniferous it is possible. Let's get a little closer. We have absolutely nothing to fear from him. For a long time it was thought to be a carnivore, but we now know that it fed on ferns, horsetails, and lycopods, plants that were common in the Carboniferous. At least that's what paleontologists think, after studying the remains of the animal, whose remains and traces of spores were found in their digestive tract. To feed itself, it uses its 2 claws and its powerful jaw. But to protect itself from predators, It is rather on its carapace with any test that it must count, and also a little on its charisma. Yet even if he is not very well armed to defend himself, he does as if. Look at him. He stands up like a cobra to impress predators. Most of the time, the subterfuge works. In the arthropod family, there is another specimen that I would like to introduce to you. If it enjoys the aquatic environment, it is still an arthropod and deserves to be on the podium of the giants within its category. Here is the Jackalopterus. It is associated with the Devonian, but there are fossils dating from the Carboniferous. This giant aquatic scorpion measures 2 meters 50 or 8 feet long. Jackalopterus renanae was the giant of its family, and according to some sources it could reach a much larger size, up to 3 meters 60 or 12 feet for the most impressive. It probably roamed lakes and rivers, and unlike other aquatic animals, it had a fairly clear and precise view of its environment. Its eyes were equipped with ommatidia, which in turn were equipped with a cylinder of lenses that refracted light and transmitted it to sensory cells. The light signals are converted into nerve signals and processed in the central nervous system. In other words, it perceives contours better, even in low light conditions, as it can be the case underwater. This is an asset to hunt its prey and to keep itself in ambush. So much for our little tour of giant insects. Did you remember why insects were so big in the Carboniferous and early Permian periods? We mentioned it briefly when we studied the giant dragonfly. The most likely hypothesis is the much higher oxygen content in the air. Some experiments have been conducted and are still conducted on the subject. The first results show that the oxygen level could have influenced their size. What about taking your mask and snorkel and going for a breath of fresh air in the open sea? We are about to start our journey in the world's oceans. This time we are in the Trias. The first animal we will meet is a carnivore. This time it is a lizardfish that awaits us. It is indeed a member of the family of ichthyosaurs. This marine reptile, which roamed the seas several million years ago now, is called Shawnosaurus sicaniensis and could reach 21 meters or 70 feet long. It is one of the largest marine animals our planet has ever known. Let's get a little closer to admire the teeth of the reptile. The teeth are quite impressive, aren't they? You should know that the diameter of their roots is 6 centimeters or 12 inches in diameter. You can imagine with such teeth, our specimen is a beautiful baby of several tons. According to the last estimates, it could reach 20 tons to be more precise. It is therefore part of the great family of the largest marine animals of all time. But unfortunately for fish and other marine animals, he was far from being the only one to hunt in the open sea. We are now entering the Jurassic period, and the predator that awaits us is just as monstrous as the one we just left. The head of this predator alone is the size of a human, about 6 feet tall. His jaw is a real killing machine. He was at the top of the pyramid, and a hunter feared by all. This presentation probably doesn't say much, but I assure you that you have to see it with your own eyes. This is the Pliosaurus. Its appearance even at the somewhat distant distance where we are, is impressive. I'll let you imagine how you would feel if you were only a few dozen centimeters away from its side. Here it is approaching. I've given you the size of his head, but I haven't gotten to the other parts of his body yet. As you can see, he is doing pretty well in the water. This is thanks to his 4 fins. The front flipper is 2 meters 75, or 9 feet long. These 4 giant fins are necessary to move the 14 meters or 46 feet long and the 50 tons of this giant of the sea. It is a real sea monster. It dominates the area and reigns supreme. No predator would dare to attack him unless he has totally lost his mind. Observe its jaws. They are formidable weapons. He is equipped to kill. The first teeth located at the front are like blades, sharp and perfectly sharpened. They are long and wide. These teeth are used to seize the prey and never let it go. The following teeth are smaller, but just as impressive. They will shred and crush the prey. Once the mouth of the Plesiosaurus is closed on its victim, the latter has no chance of getting out alive. It never misses its target. As we have seen, and we will see again, water is a privileged environment for large animals. Their morphology is often a handicap on dry land. They have to compensate this handicap with different stratagems to survive. The bearing capacity of water makes things easier for them. They move more easily. This is the case of the Plesiosaurus. The water offers him a certain ease in spite of his weight and his size. However, its extreme measurements are also its only weak point, the lack of discretion for hunting. It is visible from afar with its imposing size. The Plesiosaurus has nevertheless more than one trick up its sleeve. Its fins are long, but they are also robust and powerful. Its visibility, which could be a hindrance to bounty hunting on board, is largely compensated by the speed of action it is capable of. Experts, scientists, and paleontologists have studied the skeleton of the animal and the morphology of its fins. According to their respective conclusions, the animal mastered swimming perfectly and could probably reach a speed of 30 kilometers per hour, or 20 miles per hour. It catapulted itself into the water and swooped down on its victim by surprise. From then on, it had no time to react and no chance to escape. Mosasaurs are one of the large marine reptiles that lived in the Cretaceous period. Most of the members of this unusual family have quite exceptional dimensions, but the biggest of them is Mosasaurus. 18 meters or 60 feet long, it terrorized the seas of the planet. It is an aquatic animal which, like the whales or the dolphins that you know well, breathed in the open air. It looks a bit like a giant monitor with turtle legs and a fishtail. Its dentition leaves no doubt about its diet. It is a carnivore. He is greedy, but the least we can say is that he is not difficult. It can also throw its devotion on a turtle, a squid, a fish, or even a member of the family of pliosaurs or pterosaurs. Fierce, fast, and aggressive, no rival could compete with him on his territory. Its imposing size, its power, and its terrible jaw got the better of the rivals who were foolish enough to attack it. Let's leave the Mosasaurus to meet another aquatic phenomenon. It also lived in the Cretaceous period, but it had already conquered the sea in the Triassic period. This time, It is an aquatic animal of a completely different kind that awaits us. If it is totally different from the previous ones, I don't promise you that it will be less scary. This one lived in the Paleocene era. To all those who have a phobia of snakes, prepare yourself psychologically and emotionally for what you are about to see. Boas, pythons, and anacondas are some of the largest specimens of snakes we know today. And they are without a doubt the sworn enemies of ophiophobes. But this snake that you are about to meet exceeds in every way everything you can know or see in animal reports. Facing you is the Titanoboa. Its vertebrae are at least 5 times bigger than those of the largest anaconda species in the world today. It exceeds it largely in length, but also in width, 80 centimeters or 30 inches in diameter. Can you believe it? The circumference of its body is gigantic compared to what we know of current snakes. Its weight is just as impressive. To give you a little idea on the matter, know that the giant anaconda is one of the heaviest. Its weight can reach 250 kilograms or 550 pounds. So, with this valuable information, can you deduce the weight of the Titanoboa? It can weigh more than 1 ton. It is indeed an aquatic snake. We have not yet mentioned the length of the snake, but as you can imagine, this too is beyond comprehension. It is almost 14 meters or 46 feet long. As you have seen so far, the big problem with these behemoths is their movement. It takes a lot of energy to move, but also a skeleton and an organism adapted enough to support such a load. For our Titanoboa, the only possibility to overcome the problem of its colossal weight is water. It is only in this universe that he can move without difficulty. On dry land, things would probably have been quite different, and the movements much more complicated. Scientists believe, after numerous studies on the skeleton, but also on its diet and its environment, that the snake had to live in an aquatic environment. There is no doubt that it had no shortage of prey, and it was spoilt for choice. He was an outstanding hunter, and he did not have many predators who could match his power and charisma. His jaw was impressive. He could certainly gobble up a man as easily as you gobble up a piece of candy. This giant of the sea is terrifying. You'd think it came straight out of the worst disaster scenarios of the 7th art. However, it really existed and lived in the Miocene epoch. Its predatory instinct and its aggressiveness make it terribly dangerous. Its imposing size makes it all the more formidable. 18 meters long, or 60 feet, it weighs about 50 tons, and the opening of its mouth reaches 2 meters 10, or 7 feet. It can therefore swallow a man with disconcerting ease. The megalodon has a very particular jaw. If you are brave enough, you will be able to see by approaching a little more the opening of its mouth that this one is equipped with several rows of teeth, 250 sharp blades to be precise. Then, by observing more closely one of its teeth, you can see that it is triangular in shape, very sharp and perfectly sharpened. But that's not all. You can also see that the 2 edges of the perfectly symmetrical tooth are serrated. Each edge is like a serrated knife. They are therefore terribly effective when it comes to killing and devouring prey. It only takes a few minutes to swallow its victim completely. To give you an idea, a baby megalodon is the same size as an adult great white shark. I let you imagine what a male or a female in its prime could do. It is therefore part of the privileged category of super predators. He is at the top of the chain and fears nothing and nobody. How about a little emotional pose next to an emblematic animal of our oceans, as incredible as exceptional? Gigantic doesn't necessarily mean dangerous. If it is the case of the megalodon, the one we have an appointment with now is as harmless as a cow can be. Come on, follow me. I'll take you to meet the blue whale. It is the largest mammal in our world today, but it is also the largest mammal that has ever lived on our planet. It would have appeared during the Pleistocene. Yes, you heard right, the blue whale. If it is impressive by its size, it is also an animal that has been roaming our oceans for nearly 1.5 million years. It has crossed the millennia. It has undoubtedly met prehistoric animals that are now extinct, and it has crossed all the eras until today, from the discovery of fire to the first trip to the moon, through the Roman conquest or the discovery of the New World. In each of these eras, the blue whale was there, sailing the oceans to the four corners of the planet. Today, threatened with extinction, it is an absolutely incredible marine mammal that has its place among the giants of our planet. You know that it weighs a lot, but do you know its exact weight? It can reach 170 tons. Her tongue alone weighs 4 tons. Her heart weighs about 500 kilograms, or 1,100 pounds. You can imagine the size and weight of this organ. It is phenomenal. But these completely disproportionate dimensions are justified. A whale heartbeat lasts almost 2 seconds. Depending on its physical activity, feeding, diving, and other, its heart activity is modulated. The heart rate oscillates between 2 and 37 beats per minute. Measuring almost 30 meters or 100 feet long, the vascular and blood system of the blue whale is dizzying. The length of the cetacean's blood system is beyond imagination. In humans, the blood vessels put end to end represent nearly 100,000 kilometers or 62,000 miles. Impressive, isn't it? In our cetacean, it can reach 1 million kilometers or 620,000 miles. The arteries are so big that you could swim inside them. To maintain such a network, an organ was needed to match the task: a massive, robust, and powerful heart. You can hear the heart activity of the whale from almost 3 kilometers or 2 miles away. It is a music that must lull the little whale calf that travels alongside its mother. Although it is still very young, it is already almost 8 meters or 26 feet long. To help him grow, his mother nurses him. She has to feed well to produce enough milk. If the mother ingests more than 3 tons of krill per day, her calf needs almost 500 liters or 130 gallons of milk daily. I grant you, this figure is beyond comprehension. But when you know that the calf can take up to 80 kilograms or 175 pounds per day, you need at least that much to do it. Let's get out of the water and back on dry land. In front of you stands a strange and fascinating dinosaur. It is the Spinosaurus. I don't think there is a more surprising and frightening way to enter the Earth. Everything, absolutely everything in it is disproportionate. Its dorsal veil is gigantic. Just the spines on its back are the size of a man. About 5 meters or 16 feet high, it is more than 16 meters or 52 feet long. Its mass is also incredible. It weighs at least 22 tons. And that's not all. In addition to its extraordinary dimensions, it is also a formidable land predator. Believe me, no animal wanted to cross its path. It was avoided and feared by most of the species that shared its habitat. Its jaw, like that of the Pliosaurus we met, is equipped with different teeth all along its mouth. The first ones, longer and conical, are used to seize, the others shred, chop, grind, and finish the work. Contrary to what one might think, it was not a land hunter, but an aquatic one. Did you notice an important detail on its back legs? No? Look closely and observe its feet in particular. Yes, they are webbed. At least this is what scientists have been able to determine as the most likely hypothesis in view of the shape of its skeleton. Our Spinosaurus thus travels in fresh water. At the edge of a river, on the bank, or even deeper in a river, the dinosaur starts hunting. If necessary, it uses its back legs to move through the water. What you can't see under its thick, scaly skin is its skull. Yes, it reveals a crucial point about the predator's hunting method. The bones of its skull, especially at the front of the nose, are dotted with small holes. In crocodiles or alligators, we find similar elements on the skeleton of the skull. They are foramens. If the similarity proves to be effective, then just like crocodiles, Spinosaurus was probably able to sense the different pressures at the water level and thus detect the presence and precise movement of a prey underwater. This is a major asset for hunting, but it is far from being the only one. The great specificity of the animal, besides its imposing size, is also its capacity to hunt in water and on land. It thus multiplies its chances of survival. On land, its teeth are still as efficient, but its long, sharp, and cutting claws are a great help to seize and maintain a prey on the ground. Let the carnivore enjoy his breakfast and continue our journey on the continent. The dinosaurs we will soon see have many things in common. The first is on the time scale. They are all dinosaurs that lived in the Cretaceous period. Other well-known dinosaurs, such as the Diplodocus or the Brachiosaurus, became extinct in the Jurassic period. But the giants that we are going to observe managed for many of them to survive until the Cretaceous. The second common point of these dinosaurs is defined by their place of life. They all took advantage of the lush vegetation that covered the plains and forests of present-day South America. Among these unusual dinosaurs, we also find a third common point. They all belong to the sauropod family. We will also meet carcharodontids, which are also very impressive. But let's go back to our first group of dinosaurs. They are waiting for us with a firm foot, or rather with a heavy foot. The sauropods are the champions in terms of length and weight. They obviously have a large place in our ranking of the largest land animals that have lived on our planet. I propose you now to meet a small handful of them. Their charm should not leave you indifferent. In the featherweight category of the biggest sauropods, we find Futalognathosaurus and Alamosaurus. When I say featherweight, it is of course on the scale of a South American sauropod. In other words, we are approaching 30,000 kilograms or 66,000 pounds. Futalognathosaurus is a dinosaur that likes forested plains, such as the one right in front of us. They are herbivores and need a lot of food. You'll soon understand why. But back to our lovely Pudolnocosaurus specimen. This herbivore is known for the specificity of its skeleton at the neck level. It forms with other species a new group, that of Lognocosauria. Its pelvis measures from one end to the other more than 3 meters or 10 feet. From the cervicals and vertebrae, the pelvis and elements previously studied in cousins of this type of dinosaur, it must have been almost 30 meters or 100 feet long, counting the tail. For the weight, the estimates oscillate around 30 tons. This is about the same size as another sauropod dinosaur, Alamosaurus. This one was almost 25 meters or 80 feet long, between 26 and 30 tons. Like other members of the family, it had a long neck and a long tail. Scientists believe that it was essential for its balance, but on occasion, it was used as a whip to repel predators. As you know, Alamosaurus is also a herbivore, but take a closer look at its teeth. They are not really adapted to chewing. The most probable hypothesis is that the giant had to swallow the plant elements whole, and it is the digestive system, and in particular the stomach, which carried out the whole work of decomposition. We are now going to discover the sauropods which belong to the medium weight category, i.e., they weigh around 50 tons. In this category, we find the Patagotitan and the Nautocolossus. This dinosaur, the Patagotitan, was recently discovered, but it lived for millions of years in the Cretaceous. It far exceeds anything that has been studied so far. To give you an idea of what a behemoth Patagotitan maiorum was, here are a few details about its anatomy that should help you evaluate the size of our dinosaur. Its femur alone weighs 500 kilograms or 1,100 pounds and measures at least 2 meters 50 or 8 feet. If you add the other leg bones, hips, ribs, vertebrae, organs, muscles, and well, you are approaching 50 tons. The results are still in the order of the hypothesis, and the scientists are working on the analysis of the bones and the study supported by computer of its morphology. In any case, this titanosaur is on one of the highest steps of the podium. Its measurements are exceptional. One wonders how its anatomy was adapted to support such a massive weight and allow it to survive in its environment. How could an animal of several tens of meters or feet long and of 50 tons move with so much facility on the continent? The past is definitely full of surprises and still holds many mysteries as we speak. Come on, let's not linger. We still have a long journey ahead of us. Let's go and meet our second specimen, the Nautocolossus. Nautocolossus is another large Cretaceous phenomenon. From one of the bones, the humerus, which is still 1.76 meters or 5.5 feet long, scientists were able to estimate its size from head to tail tip. It measures a little more than 25 meters or 80 feet, and its weight is estimated at around 60 tons. An animal of this size and with such a colossal mass must have shaken the ground with every step. Imagine if they were still alive today. We already feel so small in front of an elephant, a giraffe, or a lion. How would we feel if we were to cross paths with such an extraordinary animal? Such measurements seem completely unbelievable, at the limit of our understanding, and yet the fossils speak for themselves. They tell us their story and allow us to realize how the animal kingdom, several million years ago, was completely different from what we know today. We have two more sauropods to meet. They are the biggest of all. Yes, there is an animal even bigger than the pangea titan. The Argentinosaurus could be considered as the king of the sauropods if we consider size and weight as selection criteria. The animals we have seen are already really huge, and yet one dinosaur surpasses them. The Argentinosaurus is a giant sauropod of 35 meters or 115 feet and 80 tons. 80 tons! Some scientists believe that the largest adult specimens could even reach 100 tons. One single vertebra could extend over 1 meter 60 or 5 feet high. To give you a comparison, in humans, a vertebra is about 3 centimeters or 1 inch high on average. An Argentinosaurus vertebra was the size of a 14-year-old child. If this behemoth walked around our cities, it would be as tall as a 6-story building. It would take 3 buses parked one behind the other to represent the length of the entire sauropod. From the tip of its head to the tip of its tail, the Argentinosaurus measured up to 35 meters or 115 feet long, and more than 8 meters or 26 feet high. To meet its energy needs, knowing that it is a herbivore and must therefore eat more to cover its needs, scientists and researchers have estimated the amount of food necessary for the survival of such an animal. It seems that the Argentinosaurus had a very big appetite. It had to ingest 400 kilograms, or 250 pounds of vegetation, daily. Moreover, it lived in herd. After their passage, there was not much left of the beautiful and large South American forests. His long neck was used to reach the big conifers, but he could also sweep the ground and feed on small bushes or ferns. Since it probably didn't use its teeth to chew for a long time either, it was the stomach that did all the work. Argentinosaurus sometimes swallowed stones to aid digestion. These stones are called gastroliths. Mixing the plants in the stomach with the stones allowed them to be crushed. After swallowing a few hundred kilos of vegetation, the dinosaur could go and cool off and drink a few liters of water at the edge of a river or lake. But to drink was also to take the risk of being attacked. While drinking, the sauropod is in a weak position and becomes an easy target. This is the case for most large animals, such as giraffes. Predators know this and take advantage of this time to satisfy their predatory instinct and go on the offensive. One would think that its size and stoutness were an asset against predators. No animal would dare to attack a giant, right? But contrary to what one might imagine, large sauropods were regularly attacked by predators standing in ambush. The size is imposing, yes, but it is a dinosaur exposed to all bites. If sauropods are undoubtedly the most imposing specimens in the history of our planet, other classes and families of animals deserve to make a small appearance in this journey on the land of giants. This is the case of the next dinosaur that awaits us. A little hint before meeting him? He was one of the most feared predators of the Argentinosaurus. I let you imagine what kind of dinosaur could be to be able to attack an animal of almost 80 tons. Our dinosaur of the Carcharodontidae family also lived in the Cretaceous period, along with the Argentinosaurus. It is undoubtedly one of the largest carnivorous dinosaurs known, with its 13 meters or 43 feet long, including 2 meters or 6.5 feet for the head alone. Its measurements are still only estimates, but we know that they are quite close to reality. Such dimensions, it makes you dizzy. In view of the bones found, everything led us to believe that the animal could reach about 4 meters or 13 feet high. Its femur is close to 140 centimeters or 55 inches on its own. Its weight is estimated between 6 and 8 tons, which makes it an impressive predator. It is bigger and faster than the Tyrannosaurus rex, well known by the general public. If he was faster, according to the study of his skull and the analysis of different parameters more complex, he was also less intelligent than the T-Rex. In recent years, scientists have made new discoveries and brought to light new elements about this dinosaur. They started their investigations with only 2 skulls and a few bones. Then, they discovered a 3/4 complete skeleton. We were thus able to learn more about it and confirm the hypotheses on the size and mass of this exceptional predator. Inside its skull, we can clearly see the elements that allow us to affirm the presence of very powerful biting muscles. The bite was thus formidable for the preys which crossed its way. We can also see very pronounced olfactory regions, which suggests that the predator had a very good sense of smell. It used this sense to flush out animals, then relied on its speed of action to take its victims by surprise. Gigantosaurus's teeth are just as impressive as its hunting technique. Flat and serrated, they are strong, powerful, and sharp. When the colossus grabs its prey, it slices through the flesh with ease. Many predators gnaw on the bones of their prey. Hunting parties require taking risks for one's own survival and require a lot of energy. When hunger strikes, carnivores will do whatever it takes to get food and will not waste it. They kill to eat and not be eaten. But there are also times when the bones are far too big and strong to fail under the fangs of their attackers. This is the case of our Gigantosaurus, which could not overcome the bones of the Argentinosaurus. Remember the length of a simple vertebra, 1 meter 60 or 5 feet high. A femur is nearly 2.5 meters or 8 feet long. Imagine the circumference of such a bone. No tooth could get through it. But the Gigantosaurus adapted perfectly to the situation. It favored soft tissue. After attacking the muscles or severing a tendon to prevent its victim from fleeing, it had only to bide its time. A herbivore thus weakened, without being able to move or benefit from the protection of the group, is doomed to die. Another animal hunted sauropods. These are the Mappasaurus. The Mappasaurus also hunted herbivorous colossi, but their tactics are a little different. They probably use the technique of hunting in groups. Some paleontologists have found traces of several individuals very close to each other. These elements lead us to believe that in order to defeat the largest sauropods, unity was the key to strength. Mapasaurus may not have been the largest of all predators, but it was well over 10 meters or 30 feet. Moreover, it was taking on the biggest dinosaur of all time, so it deserves its place here. The teeth of Mapasaurus are quite similar to those of Gigantosaurus. We find flat and curved teeth with a serrated edge. If this dinosaur probably did not hunt alone, but in groups, it still had to be perfectly equipped for predation. Thanks to their powerful jaws and sharp teeth, they took turns biting the herbivorous mastodon. Weakened, the bites assailed him from all sides. The sauropod did not know which side to defend himself. As a result of this attack, the sauropod finally fell. The giant gave up the game. Not being able to move anymore to eat or drink, he runs to his loss. He will die no matter what. The end will only be faster. He loses too much blood. He suffers the full force of the terrible consequences of his gaping wounds. Nor is he immune to the infections that will lead to his certain slow and painful death. For the Mapasaurus, there is no question of missing this opportunity. As he waits for the last breath of his prey, he watches for the moment when he can finally satisfy the hunger that drives him. After such a fierce and relentless fight, I think it's time for us to leave and look for a more peaceful land, don't you think? How would you like to come face to face with a Titanoceratops? The idea of a tent? Then let's go. This Cretaceous herbivore looks a lot like the Triceratops, but this one is much more imposing, with its 6 tons and 7 meters, or 23 feet high. We have mentioned much bigger and taller dinosaurs that were well over 10 meters or 33 feet. But I believe that Titanoceratops is worth a visit anyway, because one detail of its anatomy is gigantic. Here you are in front of the horn of Titanoceratops. It's quite something, isn't it? Even if it is only a herbivore, it is still impressive. He has a deep look in his eyes, and he is very charismatic. It must be said that the Titanoceratops is not quite a herbivore like the others. It has a formidable weapon of defense, its horn. It measures at least 1 meter or 3 feet long, with a circumference of 30 centimeters or 12 inches, but it could reach much larger dimensions. The head of this dinosaur is probably one of the most gigantic of all dinosaurs. Its skull measures up to 3 meters or 10 feet. That's more than a third of its entire body. In humans, our bodies are about 8 times the size of our faces. If we had the same proportions as the Titanoceratops before us, our head would be 4 times larger. There is another dinosaur that I would also love to tell you about. Just like our Titanoceratops, it is not one of the most monstrous in terms of size, but it is an extraordinary animal and sufficiently exceptional in its category to be one of the outstanding encounters of this journey on the land of the giants. If I tell you duck, you probably imagine a very small and harmless animal splashing around in the water. However, the dinosaur we are going to see has a duck's beak, and I can assure you that there is nothing puny about it. If sauropods are numerous to have become titanosaurs, they are far from being the only ones. The hadrosaurs also have their giants. In the family of hadrosaurs, there are the ornithopods, of which Magnipolia is a part. In terms of size, it is even at the top of the list. It measures about 15 meters or 50 feet long according to estimates, and can reach a weight of 23 tons. Our beaked dinosaur plays in the big league. Unlike the Titanoceratops, in the Magnipolia, it is rather the tail that is totally disproportionate. Indeed, it represents almost half of its body. If Magnipolia was equipped with a tail so long and so broad, it is because this one was a great utility to him in water. To propel such an important mass, it is necessary an adapted anatomy. The beating of the tail allows him to move in the water. It's time for you to progress a little more on the time scale. Here we are, in the Oligocene epoch. It is here that a fantastic being awaits us. When we talk about giants, we immediately think of dinosaurs. It is true that many of them are part of it, but they are not the only ones. Some have tamed the sky or the seas. Others, a little less known, have also walked the earth. These are the mammals. Look, right in front of you stands a member of the rhinoceros family without horns. Meet the Paraceratherium. This all-round champion of mammals is over 7.5 meters, or 24 feet tall, and weighs almost 21 tons. This large herbivore feeds mainly on leaves. It appreciates the open forests which facilitate its movements, and acclimatizes rather well to the sometimes wet and sometimes dry conditions. To survive, it needs to ingest large quantities of plants. It is therefore thought that the animal had to move regularly and over long distances to cover its needs. However, to move when one is more than 20 tons is not an easy task. Even a slow walk requires a lot of energy. To support such a weight, the legs of the Paraceratherium were equipped in such a way that the load is balanced and the shocks are absorbed. Look closely at the animal's front foot. You see its foot is flat, quite wide, and above all has 3 nails that look like hooves in the front of the foot. What you don't see, however, is the pad at the back of the foot. The pressure when walking is very important. The foot must compensate for this pressure to avoid pain and injury. An animal of this size, if injured and unable to move and follow the herd, is certain to die. It can no longer move to feed itself and becomes unable to defend itself. These different characteristics serve as protection. They are solid and robust bases to support its body mass and to put it in movement. But in the air as well, other specimens just as big and even more impressive have also evolved. "70 million years ago, pterosaurs, the famous flying lizards that are similar to dinosaurs, were parading in the sky. These winged lizards are a major evolution in the world of living things. If the animals reigned largely in the ocean, but also on the land, this time their fief is the sky. They are the first vertebrates to be able to move by flying. Some of them were still very clumsy or poorly equipped for flight. But over time, they were able to acquire new skills and survive, thanks to their ability to fly. If most of the pterosaurs had reasonable dimensions, others reached exceptional measurements. This is the case of the one I want to present to you now. It is admirable and deserves to open the ball of the giants of the sky. Our pterosaur is almost 5 meters or 16 feet high. To give you an idea of what this represents, the bird, if it moves on the ground in a standing position, would compete with a giraffe. Imagine a flying animal the height of a giraffe. It's hard to imagine for 1 minute that a bird could look a giraffe straight in the eye. And yet, look how incredible nature is. Here he is in the sky parading. Its wingspan, even from here, is impressive. It spans 11 meters or 36 feet, more than twice its height. Its beak is just as respectable, with its 1 meter 50 or 5 feet long. A question is probably on your mind at this moment. How could such a specimen fly, and especially take off from the surface? This is a very good question. The most experienced scientists and paleontologists in this field have also asked themselves this question. They have worked on the subject and made an astonishing discovery. Unlike some large birds that ran to gain enough speed to take off, the Quetzalcoatlus was unable to do so. So this is not the process that the giant pterosaur used. Do you have any idea? Then look, and especially observe well the technique of this giant winged lizard. He is going to make a jump, a giant leap. He manages to take off from almost 2 meters 40, or 8 feet high. Proportionally, on a human scale, that's like propelling yourself on the spot, well above your hips. The high jump is an Olympic discipline in which many athletes perform, but they need momentum, speed. The pterosaur jumps the equivalent of half its height without the momentum of speed. It is from its tendons that it draws energy. It stretches them to their maximum and then releases them with a sharp blow. His tendon works a bit like a crossbow. It stretches slowly and then releases with a snap, which causes the propulsion. When the pressure is released, the energy released is impressive. Thanks to this leap in the air, it can flap its wings hard enough to give it enough impetus to take off. This is called a propulsive vertical takeoff. Now let's see how another super pterosaur, Hatzegopteryx thambema, fares in flight. Hatzegopteryx thambema is about 13 meters or 43 feet long. If he were to walk on the ground today, he too could admire a giraffe, but he would have to tilt his beak slightly to look into its eyes. He was about 7 meters or 23 feet tall. On the ground, it is already an exceptional bird, but it is even more impressive when it opens its wings. It deploys nearly 12 meters or 40 feet of sail. It took at least one pair of wings this huge to move the 300 kilos or 660 pounds of this pterosaur. But tell me, isn't there something else that strikes you about this animal? We've talked a lot about the body proportions of some dinosaurs. Look at the size of the head in Hatzegopteryx. How long do you think it is? It's hard to estimate, I'll give you that. So I'll give you the answer. The head of this giant pterosaur is more than 3 meters or 10 feet long. In the entire history of sky animals, as far as we know, no animal capable of flying has ever surpassed the pharaonic dimensions of Hatzegopteryx thambema. Another great flying reptile invertebrate was Piscivorus, just like the Quetzalcoatlus that we met earlier. It is the Ornithocheirus. With him, the skeleton is much more adapted to the flight. It was therefore capable of some feats in the air, although, like the previous animals we met, it hovered most of the time. Its musculature is not sufficiently developed and powerful to make it fly. It took advantage of the warm air currents to move in flight. He also used another part of his body to navigate. What do you think it was? If I tell you that this part of the body is used as a rudder, and that its shape lends itself perfectly to it, does this clue put you on the way? Yes, it's the crest of its beak. Its wings were made of a membrane of thin skin, stretched and located between the front and the back legs. They could reach 5 meters or 16 feet. The hind limbs are longer and the bones are hollow, like those of our current birds, which considerably lightens the skeleton. It still weighs between 20 kilograms or 44 pounds. Besides being a pretty good pilot in the air, it was also a formidable hunter at sea. Its long jaw is equipped with vertical teeth. They are real harpoons designed for fishing. Another specificity of this pterosaur is its migration. We could see that some herbivores moved in herds, or that carnivores hunted in packs. According to the bones found in the United Kingdom, but also in Brazil, it seems that our Ornithoceras is migratory. The body of Pelagornis sandersi, which lived nearly 25 million years ago, is the size of a standing human, about 6 feet tall. We feel so small against many mammals or some reptiles, but rarely against a bird. Imagine that, if it lands next to you, it can stare you right in the eye. Its wingspan is even more impressive. Do you know the Andean condor, a vulture from the Americas, or the howler albatross, which lives mainly in the southern hemisphere? They have one thing in common: their wingspan. They are the largest birds in the world. When they open their wings, from one point to the other, a distance of nearly 3.5 meters or 11 feet separates them from each other. Yes, both would pass under a single wing of the Pelagornis sandersi, and it would take 2 Andean condors to compete with it. The Pelagornis sandersi has a wingspan of over 7 meters or 23 feet. To give you a comparison, it is the equivalent of a hang glider. Given these measurements, you might think that the weight must be proportional to its size and that it must weigh a lot on the scale. But in this case, would it have been able to fly? No, of course not. Although his size was probably a handicap, and he was neither very fast nor very agile, he was still a flying animal. It could glide over the sea and play with the currents in the sky. The study of the bones found reveals that its skeleton was hollow. This bone specificity allowed it to become considerably lighter, at least enough to fly. Scientists think that it must have weighed less than 25 kilos or 55 pounds. There are 2 things you need to remember about this giant of the sky: its flying technique and its teeth. They are both related to its survival. Let's study the first one. Its flight technique is similar to that of the howler albatross. As you can see, it hovers over the water. As you can see, it is not for nothing that it flies over the seas in this way. Its breakfast is a few meters below. The Pelagornis feeds essentially on fish. It is in particular thanks to its teeth, which are similar to those of a crocodile, that it can seize, tear up, and swallow its prey. To glide in this way above the waves, the Pelagornis uses the technique of gradient flight. This is a flight technique that allows to gain energy by repeatedly crossing the boundary between 2 air masses with distinct speeds. The lift of the air allows you to glide long enough to fish and find food. It is fantastic to see how animals have evolved and adapted to their different environments despite their imposing size. The skeleton, the musculature, the respiratory and digestive systems, or the nervous system, and the heart allow these giants to overcome the difficulties linked to their weight. The visual pathway is undeniably one of the wonders of nature. The ability to see near and far, in black and white and in color, while embracing a wide field of vision, certainly represents an evolutionary advantage of remarkable, almost miraculous magnitude. To understand the history of vision, we need to go back in time to explore the theories of eye evolution. Step by step, we understand how this organ is developed over time. Scientists have been divided into two camps. On the one hand, there are those who believe that eyes had a polyphylogenetic origin, meaning that the eye would have evolved independently several times in different species. On the other, those who believed in a monophylogenetic theory, asserting that all eyes derive from a common ancestor, and that each eye type had evolved separately according to the specific needs of the organism. According to recent studies, the trend favors the second theory, i.e., the monophylogenetic origin of eyes. Genetic research has revealed that the PAX6 gene, responsible for light detection, is common to all sighted species. The presence of all three eye types in mollusks intrinsically suggests a monophylogenetic origin of this visual organ. Exactly how long it took for these developments from a single ancestor species to occur remains unknown. Biologists have identified 4 key stages in the evolution of the eye: emergence of efficient photopigments, acquisition of directionality, formation of the photoreceptor membrane fold, and development of optical focusing ability. "600 million years ago, the first organisms developed the most primitive visual organ, consisting of a single photoreceptor, composed of 2 distinct chemical substances, a light-sensitive receptor and a pigment. Its unique function was to detect the presence or absence of light, acting as a kind of electrical switch, with only 2 possible states, on or off. The use of this structure was relatively simple, as it was not used for the perception of movement or localization like our modern eyes. Instead, its role was dedicated to a process called photoperiodicity, which was equivalent to the modern circadian rhythm. In the presence of light, certain reactions occurred, while in its absence they were inhibited. Let's meet a specimen equipped with this very first visual system. This marine animal is very primitive. The hagfish resembles an eel and has no jaws. The hagfish's eyes are very simple and resemble the pineal organs found in non-mammalian vertebrates, especially that they are positioned on either side of its head. Curiously, despite these eyes, the hagfish appears almost blind and reacts very little to light, even when its eyes are removed. It would seem that hagfish eyes aren't really made for vision. In fact, they seem to play a role similar to that of a circadian organ, i.e., they could help it regulate its biological rhythms, like our internal clock. A remarkable example of the evolution of the vision system can be seen in the developmental changes observed in the eyes of lampreys. These changes support the hypothesis that the lamprey's ancestors succeeded those of the hagfish in the course of evolution. Indeed, the Amocoetes, or lamprey larvae, have eye characteristics similar to those of the hagfish. In the larval stage, its eyes are small, sunken under the skin, and have a relatively undifferentiated retina, giving it very limited or non-existent vision. Over time, however, A slow process of neuronal differentiation takes place in a specific order. Ganglion cells, followed by amacrine and horizontal cells, then photoreceptors, and finally bipolar cells are formed. This sequence of differentiation is similar to that observed in jawed vertebrates for the development of their retina. When metamorphosis occurs over hundreds of thousands of years, the lamprey's eye undergoes significant transformations. It enlarges considerably, its retina differentiates completely, the lens develops, the cornea divides into scleral and dermal layers, allowing the eye to move relative to the epidermis. As in jawed fish, extraocular muscles develop, and finally, the eye bursts through the surface to form a visual organ similar to that of vertebrates. The process of eye development in lampreys suggests that these animals have inherited their visual apparatus from a common ancestor they share with hagfish. The existence of a hagfish-like larval eye, which transformed into a vertebrate-like eye in adulthood, supports the idea that lampreys evolved from a common ancestor with hagfish. The next step in the evolution of the eye was the fusion of these photoreceptors to form an eye spot. This advance enabled the organism to detect the intensity of light. In simple terms, if several photoreceptors were activated in an eye spot, this meant that there was strong luminosity and vice versa. This adaptation improved the perception of surrounding brightness. Once again, scientists have discovered clues in embryonic development. Early in the process, a neural structure develops and gives rise to the eye, forming two sacs, or vesicles, on either side. Later, each vesicle coils to form a C-shaped retina, which lines the inside of the eye. It's very likely that evolution followed a similar pattern. At a later stage in embryological development, as the retina folds inwards, the lens forms, leading to a thickening of the outer surface of the embryo, known as the ectoderm. The ectoderm swells into the curved space created by the C-shaped retina, gradually detaching from the rest of the ectoderm to become an independent floating element. It is likely that a similar sequence of changes took place in the course of evolution. The exact date of this change remains unknown, but in 1994, researchers at Lund University in Sweden demonstrated that the optical components of the eye could evolve relatively rapidly, within the space of 1 million years. If this is the case, it is conceivable that the image-forming eye emerged from the non-visual proto-eye in a short period of time on the geological scale. In parallel with the emergence of the eye spot, organisms also developed locomotion mechanisms such as flagella and cilia. These developments led to the need to localize the light source. To fulfill this role, several eye patches fused together to form an eye patch, like a patchwork quilt. These eye patches enabled the organism to roughly locate the light source and orient itself in its direction. By grouping several eye patches together in this way, organisms were able to improve their ability to perceive and move in relation to ambient light. In order to improve localization, the eye patch had to undergo progressive expansion. However, space was an evolutionary constraint. In response to this challenge, nature came up with a solution: the eye cup, also known as the optical cup. The development of the eye cup in vertebrates proceeds as follows. The eyes take shape from evaginations of the developing diencephalon. Depressions known as optic grooves form on either side of the rostral neural plate. As the neural plate folds upwards and inwards to form the neural tube, these growing regions project outwards and are called optic vesicles. When the expanding optic vesicle comes into contact with the surface ectoderm in a specific region called the lens placode, this induces changes in both tissues. The optic vesicle then invaginates to form the optic cup, while the ectoderm undergoes differentiation to form the lens hollow, which later transforms into the lens itself. It is sometimes suggested that the invagination of the optic cup results from the growth of the lens, but observations show that this cannot be the case. In fact, a normal ocular cup and retina can develop in the absence of a lens, indicating that these developmental processes are independent and follow distinct pathways. This adaptation enabled the organism to precisely determine the angle of the incident light beam according to the photoreceptor activated. Thus, the optic cup played an important role in improving the ability to localize the light source, despite the spatial constraints faced by the organism. By this stage, the primitive eye was fully formed, and all the species that possessed this organ entered into fierce competition for available resources. This competition gave rise to a major evolutionary event, known as the Cambrian explosion, which occurred around 540 million years ago. Scientists argue that the evolution of the eye accelerated rapidly during this period, as precise detection of prey and light gave organisms a survival advantage. So the better the eye, the better the organism's ability to survive. This selective competition for improved visual systems played a key role in the rise in diversity and complexity of animal species during the Cambrian explosion. After the Cambrian explosion, the next period saw the evolution of two essential eye structures: the iris, followed by the lens. The first step was to isolate the eye from the outside environment, which was achieved by the development of a transparent epithelial layer, now known as the cornea. Once the light detection system had been separated, the next evolutionary advantage was the ability to focus. Increased focusing power dramatically improved object localization. The first structure to develop this enhanced focusing power was the iris. Thanks to this adaptation, all incoming light was channeled into a single beam, according to the direction of the light rays, greatly improving the accuracy of light source localization. The next major development was the crystalline lens. This lens allowed light rays traveling long distances to focus directly on the retina rather than behind it. This enabled the organism not only to locate the light source precisely, but also to perceive the shape, depth, and other properties of surrounding objects in greater detail. The lens thus opened up new sensory perspectives for organisms with advanced visual systems. With the advent of the lens to capture light and focus images, the eye acquired a greatly enhanced capacity to gather information. This improvement would have generated selective pressures favoring the emergence of more efficient signal processing in the retina, going beyond the simple connections between photoreceptors and output neurons. To meet this requirement, evolution has modified the cell maturation process so that certain developing cells, instead of becoming ciliary photoreceptors, become retinal bipolar cells, which intercalate between the photoreceptor layer and the output neuron layer. This is why retinal bipolar cells bear a strong resemblance to rod and cone cells, although they lack rhodopsin and are not sensitive to light. Instead, they react to the chemical or neurotransmitter released by the photoreceptors, enabling them to play an essential role in processing visual signals before passing them on to the output neurons. Camera-type eyes offer a wide field of vision, usually around 180 degrees. In practice, however, our brains are limited in their ability to process all the visual information available at any one time, due to the limited number of nerve fibers connecting our eyes to our brains. As a result, we can only sample a fraction of the visual data available. It is likely that the first camera-like eyes faced an even more severe limitation, with probably an even smaller number of nerve fibers. This constraint would have exerted considerable selective pressure in favor of the evolution of muscles to move the eyes. The presence of such muscles must have existed around 500 million years ago, as the arrangement of these muscles in lampreys, whose lineage goes back that far, is almost identical to that of jawed vertebrates, including us humans. Despite the ingenious features of vertebrate eye evolution, a number of rather counterproductive traits can be identified. For example, the retina is positioned upside down, which means that light has to pass through the entire thickness of the retina, including nerve fibers and intermediate cell bodies that scatter light and impair image quality before reaching the light-sensitive photoreceptors. In addition, blood vessels cover the inner surface of the retina, casting unwanted shadows on the photoreceptor layer. Another tricky aspect is the presence of a blind spot, where nerve fibers that cross the surface of the retina come together before tunneling through it to emerge as the optic nerve at the back of the eye. This list of inelegant features could be extended to include other examples. These imperfections are by no means inevitable features of a camera eye, as octopuses and squids have evolved independently of camera eyes which do not have these defects. Examining the vertebrate eye in an evolutionary context highlights these seemingly absurd shortcomings as the consequences of an ancient sequence of evolutionary steps, each of which brought advantages to our vertebrate ancestors, long before they developed advanced vision. The eye has evolved in many different ways over time, giving rise to a variety of features in different species. Jellyfish, for example, have eyes, and although they have no brain, these eyes transmit information directly to the muscles for rapid reaction. Nautiluses have an aperture without a crystalline lens, adopting a different focusing mechanism. Flies, on the other hand, have many eyes, giving them a wider field of vision. The diversity of ocular adaptations continues with predators, which have forward-facing eyes for better depth perception, while prey have lateral eyes, offering a wide field of vision for detecting potential dangers. In the case of humans, they have developed the ability to artificially correct their vision using devices such as spectacles, enabling them to compensate for certain visual imperfections. Ultimately, the list of ocular adaptations is truly infinite, with each species evolving uniquely to suit its environment and specific needs. All the auditory organs of today's terrestrial vertebrates, whether reptiles, birds, or mammals, have evolved in completely distinct ways. Ciliated cells, the sensory cells at the base of the auditory organs of all vertebrates, emerged very early in the evolutionary history of animals. They play a significant role in today's non-vertebrate animals, such as ascidians, and can be traced back to the first animals with well-developed tissues, the cnidarians, like sea anemones. The auditory part of the ear evolved from a vestibular organ whose evolutionary origin, in particular, the sensory cells themselves, resembles that of the lateral line, a body surface organ used by fish to detect the movement of fluids in water. The lateral line has sensory hair cells similar to those in the cochlea, whose responses are strongly influenced by viscous damping and are linked by a gel-like structure. The damping problem was solved early on by evolving the active mechanisms of the sensory cells and linking them via a gel-like structure to increase both their sensitivity and frequency selectivity. These two features have survived 500 million years of evolution and are common to vertebrate inner ears. During the Carboniferous period, from -359 to -299 million years ago, tetrapod ancestors successfully migrated to terrestrial life, paving the way for further adaptations of existing sensory systems, including hearing, from aquatic to terrestrial life. Thus, over hundreds of millions of years, progressive adjustments were made to these organisms so that they could adapt to higher frequency stimuli, first in an aquatic environment and then in an aerial one. It is assumed that the ancestors of amniote vertebrates had a limited hearing capacity for loud, low-frequency sounds, as they lacked a tympanic middle ear. The first terrestrial vertebrates had an auditory papilla that rested on solid tissue and was covered by an otolith, a small mineral structure present in the inner ear of certain animals, notably fish and reptiles, responsible for detecting movement and balance, and composed of calcium carbonate crystals. In amphibians, the otolith membrane was replaced by a tectorial lining that contained no otoliths. In all subsequent lineages, the basilar membrane appeared, freely suspended. In other lines, the basilar membrane showed some adaptation, with moderate frequency selectivity. Around 100 million years after the separation of the tetrapod lineages during the Triassic period, at least 5 independent developments of tympanic middle ears were observed. These developments functioned as essential impedance matching mechanisms for the efficient detection of airborne sounds. The absence of an effective middle ear prevents the detection of high-frequency sounds. This gave rise to parallel evolutions, leading to major differences in the auditory systems of tetrapod vertebrates between different clades, while ultimately fulfilling the same function: detecting, transmitting, transducing, and encoding airborne sounds. Paleontological, morphological, and developmental evidence suggests that lepidosaurs, such as lizards and snakes, as well as archosaurs, which include birds and crocodiles, independently developed a middle ear, composed of a single ossicle. Firstly, in birds and crocodilians, the structure of the auditory organ shows marked similarities, such as the presence and distribution of similar cell types. It is therefore reasonable to assume that this structural configuration was reached or initiated at the latest around the separation of their ancestral lineages some 160 million years ago. Similar reasoning applies to lepidosaurs. In Tuataris, which separated early from other lepidosaur lineages, the cellular configuration of the auditory papilla is similar than in lizards. In Tuataras, as in turtles, there is only one type of cylated cell, and all cylated cells have the same orientation. Thus, the more complex structure typically seen in lizards seems to have arisen after the Tuatara lineage separated from the squamates. However, it is not known whether lepidosaurs, including the Tuataras, developed a tympanic middle ear all at once. Finally, in mammals, the characteristic structure of the auditory organ, known as the organ of Corti, is essentially found in all recent groups of mammals, although there are marked differences between monotremes and therian mammals. We can therefore assume that the organ of Corti appeared before these lineages split, between 220 and 150 million years ago. This comparative analysis suggests that the evolution of the tympanic middle ear in each lineage had significant implications for the later development of the auditory organs of the inner ear. The reduction in bone size and the emergence of new configurations to form the tympanic middle ear probably improved not only auditory transmission in general, but especially that of higher frequencies. This new feature undoubtedly exerted additional selection pressures on the auditory organ to enable optimal transduction of information and its transmission to the brain. Over time, the mammalian cochlea evolved specifically in different subgroups. Over the course of several subsequent geological periods, mammals diverged considerably, and although the basic structure of their cochlea was relatively constant, differences in length and various specializations emerged. Not only did cochlea develop huge differences in length, they also developed specific specializations in groups pursuing different strategies for locating prey, using ultrasonic frequencies in the air, such as bats, and high ultrasonic frequencies in the water, such as toothed whales. For example, some bat species have developed cochlear regions where the length of the organ of Corti, dedicated to specific narrow frequency ranges, is considerably enlarged. These adaptations have enabled bats to better detect and localize sounds in their environment, improving their hunting and navigational abilities. On the other hand, there is no reason to expect mammalian auditory organs to be necessarily superior or more sophisticated than those of other groups. Each present-day vertebrate group has developed an appropriate auditory solution according to the evolutionary pressures dictated by the environment, predator-prey relationships, and intraspecific communication needs. Acute sensitivity and precision in frequency resolution, characteristics commonly associated with mammalian auditory organs, have evolved independently in all groups of modern terrestrial vertebrates, in response to their own specific evolution and adaptation. Indeed, some species of lizard possess auditory nerve fibers tuned to some of the finest frequencies of any vertebrate, including most mammals. Birds of prey, particularly owls such as the barn owl, rely almost exclusively on hearing to locate their prey, so auditory sensitivity exceeds that of most mammalian species. Echolocating mammals such as bats and dolphins exhibit a wide range of hearing frequencies, a characteristic almost equaled by certain species of frog, which use ultrasonic calls to communicate. In humans, although generation times are 10 times longer than in fish, the issue is not so much the creation of hundreds of new species, but rather the adaptation of a sensory organ to optimize the processing of communication signals vital for survival and hence reproduction. Whether the evolution of the human cochlea paralleled the development of language is a fascinating question that could be explored by future research. All living cells are irritable to certain chemicals, and this predisposition of cells to be perturbed by chemicals is likely to have led to the eventual evolution of specific receptor proteins to detect chemical signals, and ultimately to specific chemosensory organs and systems, such as the sense of smell. Olfactory capacity, used for chemical communication, is widespread in the majority of living beings belonging to the animal kingdom, enabling the perception and identification of chemical signals. The sensitivity and chemical range of animals' olfactory systems are remarkable, giving them the ability to detect and distinguish between thousands of different odor molecules. In the animal world, olfaction has evolved to enable the detection of signals from the chemical environment, which contains decisive information on the direction to follow, food sources, the right time to reproduce, as well as stimuli to consider pleasant or dangerous. An animal's reaction to chemical signals is the result of a combination of mechanisms acquired during its lifetime and innate predispositions resulting from the cumulative impact of individual experience and evolution through the generations on the olfactory nervous system. Evolution has shaped the olfactory system in response to two challenges: the need to explore a wide range of chemical information and the specific sensory challenges faced by each animal species. Generally speaking, vertebrates develop an olfactory system that detects odorants and pheromones through their interaction with cell surface receptors specialized on olfactory sensory neurons and organized in dramatic and subtle ways, enabling adaptations in response to major changes in environment and lifestyle, such as the transition from ocean to land and the predominant development of vision. In terrestrial vertebrates and insects, we often think of olfaction as a chemosensory modality dedicated to the detection of low concentrations of airborne volatile chemicals. Yet aquatic fish and crustaceans, while not encountering airborne volatile odorants, possess sensory systems anatomically similar to the olfactory systems of terrestrial animals. For these aquatic animals, odors are sapid molecules in solution. Olfaction is therefore not necessarily the detection of volatile molecules in the air. In this context, it has been suggested that during the transition between aquatic and terrestrial environments, new demands emerge for chemosensory systems linked to odor detection. This happened in a remarkable way, as olfactory stimuli evolved from hydrophilic to predominantly hydrophobic and volatile compounds. This transition granted terrestrial vertebrates an evolutionary option to detect volatile water-insoluble odorants. As a result, it is thought that the complex biosynthetic pathways required to produce volatile compounds, as well as the highly complex combinatorial coding that characterizes olfactory perceptions, driven by olfactory receptors, emerged suddenly in just a few 100,000 years. Comparative genomic and phylogenetic studies argue that chemosensory receptors were conserved across aquatic and terrestrial taxa, and that mammalian-like olfactory receptor genes evolved in marine environments before the first vertebrates appeared. These changes in olfactory receptors at protein level, altering their interaction with odorant molecules, play a key role in the evolution of olfactory perception. However, researchers have discovered that this adaptive evolution of olfactory perception can also be influenced by co-expressed olfactory receptors undergoing changes in copy number and protein sequence. Selection also acts on individual olfactory receptor families or subfamilies, favoring specialized adaptation or facilitating chemical communication between members of the same species. Finally, genetic variation among the thousands of unique olfactory receptor genes can lead to behavioral modifications in response to specific chemical signals. Thus, the combination of these mechanisms enables vertebrates to adapt in precise and complex ways to their changing chemical environment. An animal's genome evolves in such a way as to be closely linked to the specific tasks resolved by its nervous system, enabling it to detect odors and respond appropriately to promote survival and reproduction. Recent studies have revealed the presence of ancestral versions of synaptic genes involved in fundamental neuronal communication in various organisms without a nervous system, suggesting a progressive evolution of olfaction. In parallel, studies have clarified that the olfactory capacities of multicellular organisms, as well as chemosensory learning abilities, evolve from life forms not equipped with complex neural networks. In particular, prokaryotes have been proposed as model systems for understanding the evolution of olfactory systems in higher organisms. In fact, bacteria respond to volatile chemical gradients, but also use contact-dependent signaling. In addition, a unicellular but multinucleate eukaryotic organism, Viscerum polycephalum, has been shown to be capable of chemosensory detection. Studies have shown that olfactory receptor cells are primary bipolar neurons with a similar morphology between species. They have filamentous dendrites to increase the surface area for stimulus capture, and an axon that extends to the central nervous system. Dendritic processes are mainly of ciliary origin in most animals. Although the overall morphology is conserved, the fine details of olfactory receptor cell morphology can be adaptive, i.e., they can vary in a habitat-dependent rather than species-dependent manner. For example, the olfactory receptor cells of land-dwelling crustaceans, such as the giant thief crab, have a morphology closer to that of insects than to that of marine crustaceans. Crustaceans share a common ancestor with insects, and as they lack olfactory receptors, it has been proposed that these receptors evolved when prehistoric insects left the sea to live on land. According to this idea, olfactory receptors evolved because these ancestors must have been able to detect odorous molecules floating in the air, rather than dissolved in water. But a recent discovery contradicts this hypothesis. The discovery of a new type of olfactory receptor, ionotropic receptors, IR, has changed the order of things and opened up new avenues of inquiry into the evolution of olfactory receptors throughout the history of life. These new olfactory receptors are present in crustaceans, in which no classical olfactory receptor had been found until now. Since crustaceans were the first to appear in the history of life, before insects and before vertebrates, we can assume that these IR were inherited long before the appearance of the first insects. During development, the olfactory system takes shape from the olfactory placodes, specialized regions of the anterior ectoderm that share cellular and molecular characteristics with other placodes involved in the development of various cranial senses. Certain cell types of the olfactory system preceded the appearance of vertebrates, as did certain mechanisms involved in placode formation. It is likely that these two elements were already interconnected in the common ancestor of vertebrates and tunicates. In vertebrate ancestors, this system evolved into a more complex organ system, integrating additional tissues and specific morphogenetic processes to develop distinct olfactory components. Subsequently, the ancestral placode divided to give rise to the paired olfactory organs characteristic of most modern vertebrates. Thus, a major olfactory adaptation occurred along the amphibian and terrestrial branches of the vertebrate tree, resulting in the formation of distinct neuroanatomical structures expressing more recently derived olfactory receptors. Aquatic vertebrates such as fish and amphibians have an olfactory system similar to that of terrestrial vertebrates, but specialize for the detection of chemicals present in water rather than air. Insects, on the other hand, have a well-defined system in their antennae for detecting airborne chemicals, generally referred to as the olfactory system. However, this system is evolving convergently, independently of vertebrates, to perform this function. Unlike fish, amphibians have developed a separate vomeronasal organ, which specifically detects attractive sex pheromones specific to amphibians. In addition, they possess an air-filled main olfactory epithelium, which expresses a vast repertoire of novel Class II olfactory receptors that have acquired a role in innate reproductive behaviors. These observations suggest that odor interpretation depends not only on the odorant molecules detected, or ligands, but also on the developmental identities and specialized sensory pathways of the cells that detect these odors. Recent evidence shows that individual mammalian olfactory receptor cells can be activated by several different odorants, and that individual odorants activate several receptor cells expressing different receptor proteins. This combinatorial coding is also present in insects and fish, suggesting that it has been conserved in the evolution of olfaction. Consequently, there are striking similarities between species in the organization of the olfactory pathway, from the nature of olfactory receptor proteins to the organization of the olfactory central nervous system. Although there is a striking evolutionary convergence towards a conserved organization of signaling pathways in vertebrate and invertebrate olfactory systems, the receptor gene families involved have evolved independently. These common organizational features span a wide phylogenetic range of animals. Such conservation also implies that there is an optimal solution to the problem of odor detection and discrimination. Either this solution evolved relatively early and was then retained in evolution, or more likely, animals convergently developed identical or similar solutions to the odor detection and recognition problem. The sense of taste is a specialized chemosensory system, one of the main mechanisms used by animals to assess the nutritional quality of food, helping them to make decisions about consuming beneficial foods while avoiding harmful substances in their daily lives. Unlike touch, vision, hearing, or olfaction, which are called upon in different behavioral situations, the sense of taste has evolved to play a primordial role in food regulation and motivation. Taste systems detect essential nutrients, as well as harmful substances in food, and trigger innate behaviors that lead to the acceptance or rejection of potential food sources. Most vertebrates can perceive 5 basic taste modalities: bitter, umami, sweet, salty, and acidic. All vertebrates have some ability to avoid harmful chemicals and seek out nutrients. It is thought that each sense of taste has evolved to detect and motivate the consumption of essential nutrients, and also to detect and avoid potential poisons. It is widely accepted that sweet taste evolved in plant-eating animals to detect energy-rich simple sugars, such as glucose, fructose, and sucrose. Bitter taste, on the other hand, probably functions to ensure that an animal avoids poisons. Indeed, most poisons are bitter, and most bitter substances are harmful, although this relationship is not perfect. The salty taste allows the detection of sodium, an absolutely essential mineral. When certain species of animal lack sodium, and this usually occurs in herbivorous animals, a powerful appetite for salty taste is aroused. And for many species, salt is consumed even when there is no apparent need. As for the sour taste, many have suggested that it is involved in detecting fruit ripeness. Finally, the 5th basic taste, umami, probably serves to signal amino acids and proteins. By comparing animals from different branches of the evolutionary tree, researchers have deduced that the sense of taste probably evolved over 500 million years ago. This would have occurred before the divergence of terrestrial vertebrates, bony fish, sharks, and lampreys, when their common ancestor, a primitive fish, developed a new type of cell. These chemosensory taste receptor cells, located in taste buds, are able to detect chemical stimuli and send signals to the brain via the taste nerves to generate taste sensations. Taste buds are the gustatory end organs in vertebrates ranging from lampreys to mammals. These end organs respond to a variety of sapid chemicals and transmit signals to afferent nerve fibers from 3 cranial ganglia: facial, glossopharyngeal, and vagus. Over time, taste buds have undergone several modifications to adapt to the dietary needs of different animal species, which explains their varied location in these animals. Not only are they found in the mouth and pharynx, but they can also be widely distributed across the surface of their bodies. Evidence suggests that umami receptors were the first to develop. Researchers have reported the discovery of genes similar to those encoding the receptors used by humans to detect the amino acid glutamate in the genome of the elephant shark, a species that split off from other fish 400 million years ago. Sharks lack bitter taste receptors, suggesting that these genes evolved more recently. Ancient fish were probably the first vertebrate animals capable of detecting sour taste. It is likely that these fish did not taste food with their mouths, but rather could detect acidity in the ocean using the outside of their bodies. Variations in the carbon dioxide dissolved in water can create different levels of acidity, which can be dangerous for fish. So the ability to sense acidity would have been essential for their survival. Bitter taste is perhaps the taste that most intrigues evolutionary biologists. It's the Darwin's finch of taste, engineered and customized to suit a species' ecological niche. Humans have 24 or 25 different bitter receptors, depending on the individual, each recognizing unique combinations of chemicals. Toxic bitter compounds come in all shapes and sizes, so it makes sense that the receptors that recognize them are diverse. The evolution of bitter taste, therefore, also involves distinguishing between different chemicals. The diets of humans and their ancestors are mainly based on plants, which are an abundant and accessible source of nutrition. However, plants have developed physical, reproductive, and chemical defenses to protect themselves from herbivores. Herbivores must therefore contend with various obstacles such as thorns, shells, and symbioses with defending animals. Herbivores have developed various strategies to overcome the toxic defense mechanisms of plants. Among these, a particularly effective and widespread method in vertebrate animals is the detection of bitter flavors. It has long been widely recognized that many plant toxins are perceived as bitter, creating a powerful mechanism for minimizing exposure to these harmful substances. By signaling the presence of toxins in foods before they are fully consumed, bitter taste detection helps avoid their ingestion. This ability plays an essential role in the survival of herbivores, helping them to identify and avoid potentially dangerous plants. The ubiquity of bitter perception in vertebrates and its complex role in humans have raised questions about its evolution. One of the most remarkable revelations of vertebrate comparisons is that bitter taste receptors are a relatively recent evolutionary innovation. While they are present in bony fish, lobe-finned fish, and tetrapods, they are not found in cartilaginous fish. Thus, bitter taste receptors must have first appeared in an aquatic species around 430 million years ago. This makes them the most recent of the chemosensory receptor families in vertebrates. The origins of the receptors involved in taste perception in animals are the subject of much speculation. Similarities between the evolution of vascular plants and the Cambrian explosion in animals suggest that the origin of these receptors may be linked to an important transition in plant to herbivore interactions. However, there is also evidence that these receptors play other roles, such as detecting nutrients in the gut, suggesting that their function as taste receptors may have emerged later. The bitter taste receptor family has diversified within and between taxa, with repertoires of bitter taste receptor genes varying in size across species genomes. Studies show that selective pressures on taste perception represent the most obvious potential driver of diversification of these genes. Fish have different numbers of genes, ranging from 1 in Amazon molly to 74 in coelacanth, but most fish have an intermediate number of genes. Variation in the number of bitter taste receptor genes occurs in different groups of animals, such as fish, amphibians, and saurians. Caecilian amphibians, which include limbless amphibians, have the lowest number of genes, while anorans, which include frogs and toads, have the highest number among all recorded vertebrates, probably reflecting their adaptation to variable lifestyles and environments. In fact, most frogs and toads inhabit both aquatic and terrestrial niches, which necessarily contain a greater variety of toxic substances. Consequently, their ecological needs should encompass very different requirements for their gustatory systems. For example, aquatic factors such as pH have been shown to influence the divergence of taste receptor genes. Most frogs feed on worms, insects, and other small arthropods, which contain more potentially toxic substances. It is known that insects often sequester toxic compounds from plants for their own defense, suggesting that anorans may benefit from the ability to detect dangerous prey. While caecilians are animals adapted to life underground, living in soil layers rich in organic matter, they build tunnels to get around. All caecilians are carnivores, feeding on earthworms, plathelminths, arthropods, frog eggs, and tadpoles. Compared with frogs, caecilians are less sensitive to bitter taste, as animal tissues contain fewer toxic substances. Consequently, we hypothesize that the lifestyle, ecological, and dietary complexity of amphibians raise their evolutionary pressure for a wide variety of taste receptor genes. Saurians, which include crocodiles, lizards, snakes, and birds, also show variation in the number of these genes. Snakes have low numbers, while crocodiles, turtles, and lizards have higher numbers, similar to many fish. Repertoires of bitter taste receptor genes in birds and mammals show great diversity. Penguins and cetaceans have few or no functional genes, while birds such as zebrafinch and white-throated sparrow have several. Primates, including man's closest relatives, have an average number of genes, with the gibbon having the fewest and the chimpanzee the most. Although toxins are a threat to all herbivorous animals, not all species are equally exposed. This variability is reflected in the size of genetic repertoires, which is related to the proportion of plant-based diets for each species. This trend is particularly marked with herbivores having larger repertoires than carnivores. The most extreme example of this variation is found in obligate carnivores such as whales, penguins, and pinnipeds, which swallow their food without chewing, resulting in a total absence of certain genes linked to toxic detection. Around 7 to 8 million years ago, humans had one last common ancestor with other great apes. If we look at the current eating habits of great apes, we can deduce the diet of our common ancestor. It seems that this species was omnivorous, feeding mainly on tropical fruits, and also included leaves and insects in its diet. Chimpanzees, our closest relatives, rely heavily on fruit for their caloric intake, with a small proportion of their diet coming from animals such as other apes and insects. Between 4.4 and 2.3 million years ago, human ancestors left the forest for the savanna and other ecosystems, and their food repertoire expanded considerably. Thus, over time, early hominids gradually abandoned the eating habits of forest monkeys and increased their ability to obtain a variety of nutrients in order to adopt a more diversified outdoor diet. Despite this dietary evolution, our need for fruit and our attraction to sugars and acids, shared with other great apes, remained intact. From a nutritional point of view, fruit is attractive because of its sugar content, which provides intrinsic satisfaction, as well as vitamin C, which is essential for life support in hominoids. The question of why humans are sensitive to acidic flavors and even inclined to prefer acidity has sparked debate in the scientific community. Signals from acidic substances do not provide much nutritional value, with a notable exception of vitamin C. However, this exception is significant because, unlike most mammals, monkeys and apes are unable to synthesize vitamin C due to the loss of a functional version of the gene encoding gluconolactone oxidase. The common ancestor of arthropods that suffered this enzymatic loss probably had to rely on a sufficiently rich source of ascorbic acid from fruits and other plants for the enzyme to become useless. It is plausible that the acidic taste played a necessary role as a guide to identifying vitamin C-rich fruit. In addition, the combination of acids with sugars could also have been used to assess the degree of ripeness of the fruit, using mixtures of sweet and sour flavors. Thus, acids are not stimuli for which we have evolved to react in isolation, but rather elements we have experienced in the context of the sugars present in fruit. As a result, sweet and sour flavors are perceived as synergistic in the fruit aroma palette. In addition, acid and sour taste act as indicators of fermentation processes which human beings around the world seek out and consume. Wild apes depend mainly on forest plants, especially young leaves, for most of their daily protein intake. Nevertheless, early hominids and modern humans have shown a preference for consuming slightly more digestible protein sources, such as meat. Although umami taste is not immediately apparent in fresh meats, it develops significantly in aged or cooked meats. Consequently, it seems that chimpanzees do not possess a gustatory system specifically dedicated to detecting the taste of glutamate or ribonucleotides, even though they can perceive these stimuli. The characteristic umami taste of hydrolyzed protein is mainly carried by glutamate and ribonucleotides. Humans have developed a preference for glutamate, ribonucleotides, and umami taste, probably as signals indicating more easily digestible proteins in lightly aged or cooked meats. Indeed, there is ample fossil evidence to suggest that culinary practices predate the advent of modern man. Humans the world over take advantage of a wide variety of plant and animal products that undergo fermentation. Our strong attraction to the taste of free amino acids and ribonucleotides may stem from a tendency to consume fermented foods, including lightly aged and/or cooked meats. This category of food would bring various benefits to the survival of our species. Fermentation facilitates access to macro and micronutrients while providing probiotic bacteria. Although it has been assumed that the pleasant taste of glutamate and ribonucleotides acts as an indicator of protein, many protein-rich foods are not particularly tasty or umami when fresh. It's the process of fermenting or aging these foods that releases the umami, savory taste of protein. Thus, our attraction to amino acids, particularly glutamate, as well as salty taste, may have emerged from our desire for fermented foods, taking advantage of the nutritional improvements and benefits of probiotic bacteria for our species. Humans stand out from other mammals thanks to a unique phenomenon, a large-scale polymorphism of the salivary amylase gene. This change in our genetics could be the result of our past history linked to the consumption of starch-rich tubers, and could have occurred around 100,000 to 200,000 years ago. The number of copies of the salivary amylase gene present in our genome directly influences the production of amylase in our saliva. This enzyme in high quantities and efficiency has the ability to rapidly transform a cooked starch, such as a thick pudding, from a semi-solid to a liquid state in a matter of moments in our mouths. The resulting sugar concentrations can produce sufficient substances such as maltose, which activate brain regions linked to taste and reward. In short, it seems that the human mouth harbors a taste receptor that responds to glucose polymers without necessarily inducing a conscious taste sensation. This receptor can be stimulated by the products of starch decomposition in the mouth, especially in the presence of high levels of salivary amylase. Human beings have a natural tendency to seek out the taste of salt in food. We find the taste of moderate concentrations of salt very appealing, as do other omnivores. But high levels of salt are considered unpalatable to us, as they disrupt the osmotic balance of our body fluids. Salt is a widely used ingredient in food preparation throughout the world, and individuals from different cultures consume roughly the same amount of salt every day. This similarity in salt consumption across cultures suggests that our salt intake is regulated by intrinsic biological factors. A carnivorous animal absorbs salt at every meal, while a herbivore can easily run out of sodium, triggering a desire for salt, which translates into a search for natural licks. An omnivore's exposure to dietary salt would therefore logically be intermediate between that of carnivores and herbivores. It should be noted, however, that humans lose salt through perspiration, which may explain why we have a slightly higher salt intake preference than other omnivores. Since our earliest prehistoric ancestors, the sense of touch has been central to our survival and reproduction. What we touch can have a direct impact on our ability to survive and reproduce. It may be a predator, prey, or parasite, a parent, lover or child, shelter or danger, a tool or food source, the ground beneath our feet, or the branch we use to swing. It's also the first sense to develop, with some fetuses responding to touch in the womb after just 8 weeks. Evolution has created a tactile system made up of different sensors and receptors, which together detect the qualities of what touches our body and what our body touches, such as weight, temperature, and texture, so that we can deduce useful information. Many animals, including some coelantherates, annelid worms, Insects and many other arthropods, birds and mammals, have hairs or capillary projections that are richly supplied with nerves and serve to indicate to the animal that it is in contact with an object. These hairs may have been specially modified, for example, vibrissae or whiskers in certain areas of the body, such as the face or toes, to provide more sensitive discrimination between stimuli. A second type of tactile receptor, the subcutaneous receptor, is found in the skin. Receptors of this type are found in almost all animals and may consist of free nerve endings or complex corpuscles. Unicellular organisms already possess the capacity for directional detection of tactile stimuli, as can be seen in ciliates such as paramecium, which move away from the stimulus source following mechanical disruption of their outer membrane. Sponges, the simplest multicellular animals, do not possess neurons, but they do have the capacity to respond to variations in water flow and pressure in response to the movement of their immobile cilia. However, the evolution of neural conduction has led to a radical change in the ability to respond rapidly and flexibly to tactile stimuli. Cnidarians such as jellyfish and hydra, despite their relatively simple nervous systems, can exhibit coordinated patterns of motor response to sensory stimuli, and many have a rich capacity to respond to touch. For example, hydra nematocytes are structures that provide a good model for understanding the mechanoreceptors of more complex invertebrates. In jellyfish, groups of hair cells called tactile combs regulate complex behaviors, including escape, feeding, and locomotion. The advantages of sensitivity to mechanical stimuli provided by hair-like structures may have encouraged their convergent evolution in several animal lineages. For example, the ciliated cells of jellyfish appear to be sufficiently different from those of vertebrates that a common origin for both is unlikely. Studies into the molecular basis of mechanosensation in different classes of animals also suggest that the cellular mechanisms required for tactile detection may have evolved several times. All animals with a central nervous system respond to touch. The nematode, Sanor habditus elegans, is an important model organism in neuroscience. It is the first animal system in which the entire neural network involved in tactile behavior has been identified. This worm has mechanosensory cells located in the integument beneath the cuticle, linked to 6 sensory neurons, 10 interneurons, and 69 motor neurons. In larger, probably more advanced animals, touch-induced behavior becomes more versatile and complex. The leech has developed a central nervous system with a succession of ganglia for each segment. Each ganglion houses 3 groups of mechanosensory neurons, all of which respond to mechanical stimulation of the body, but with different response thresholds. Tactile cells are the most reactive and respond to the gentle touch of the body wall. Pressure cells respond to more intense tactile stimuli, while nociceptive cells are sensitive to very powerful and potentially harmful stimuli. In arthropods, mechanoreceptors are located in strategic areas of the body where movement is most likely to occur. In proprioception, these are the joints, muscles, and tendons. In contact, they are the surface areas where contact with external objects is most likely. Arthropods exploit these strategically important locations in 2 ways. On the one hand, they use tufts of hairs called capillary fields or capillary plates to detect movement between adjacent body segments through proprioception. On the other hand, to detect contact, they use similar hairs but located in different parts of the body, with a particularly high concentration where contact is most likely and/or most important to detect. Spiders and insects also use hair sensilla for proprioception and consequently for active touch detection, where the active movement of a limb must be monitored. Among today's arthropods, only myriapods, crustaceans, and insects have specialized touch sensory structures, the so-called antennae. These sensory organs are equipped with a particularly large number of sensillae, displaying a density far greater than that found on most other body regions. Crustaceans have 2 pairs of antennae, while insects have only 1 pair. The generally accepted hypothesis is that over the course of evolution, insects are likely to have lost their 2nd pair of antennae, meaning that the insect antenna shares a common evolutionary origin with that of crustaceans. Fish and amphibians have mechanoreceptive organs called neuromasts, which are arranged in rows on their body, head, and tail. These neuromasts may be present autonomously on their skin or inside fluid-filled channels. The main sensory component of each neuromast is a group of hair cells that respond to hydrodynamic stimuli caused by water movement or variations in water pressure. In this way, the lateral lines provide a sophisticated sensory system for detecting the distant origins of water movement, such as predator or prey behavior, water currents, and the topographical features of the underwater environment. Mammalian skin evolved from the integument of earlier vertebrates in a complex evolutionary process that is still only partially identified. What is clear is that the first mammals, or perhaps their reptilian, therapsid ancestors, redeveloped hair as an adaptation of the outer epidermal layers of their integument. While dense hair clearly has a thermoregulatory function, the evolution of hair in mammals cannot be attributed solely to the goal of regulating body temperature. On the contrary, it is very likely that the first hairs were primarily used for tactile functions, and their subsequent evolution gradually gave them a secondary role as insulators. The main sensory function of hairs persists in vibrissae, also known as tactile hairs, present in all therian mammals, such as marsupials and placentals, with the exception of human beings. In cases such as the naked mole rat, a species that has lost its entire coat, tactile hairs have been maintained throughout the body because of their importance to the sense of touch. Many mammals have evolved to have areas of hairless skin, also known as glabrous skin. In humans, these areas include the skin of the lips, hands, fingertips, and soles of the feet. These are the parts of the body that play a key role in physical interactions with the outside world, and where tactile precision is particularly important. So it's not surprising that hairless skin is characterized by a high density of mechanosensory receptors that are extremely sensitive to delicate touch, which could contribute to the effective or social tactile capacity that could be specific to mammals. Only mammalian species have elongated facial vibrissae, also known as vibrassae, which extend outwards and forwards from the animal's snout. They form a tactile sensory network that envelops the animal's head. Species without vibrissae, or in which they were less apparent, were generally the most evolved representatives of their order. Consequently, having facial vibrissae was an ancestral characteristic of mammals. Many terrestrial mammals, especially those that are nocturnal, live in low-light environments or climb trees, and have evolved highly specialized tactile sensing systems via vibrissae. Research has focused on a variety of these animals, including rodents such as the rat, mouse, and golden hamster, as well as insectivores such as the Etruscan shrew and the Brazilian short-tailed marsupial possum. In some seal species, vibrissae have adapted to detect disturbances left in the water by fish. By measuring these hydrodynamic flow fields, these animals are able to track and capture fast-moving prey in total darkness. Deep-sea foraging pinnipeds, such as the bearded seal and walrus, have evolved a highly specific vibrissal adaptation designed to optimize their efficient search for shellfish and other invertebrates in the muddy layers of the seabed. The abundant, dense, and mobile vibrissae of these animals combine to form a kind of sensitive, tactile rate that enables them to detect and ingest their prey at exceptionally fast speeds. The Ordovician period is divided into 3 eras with 7 distinct stages. From 485 to 471 million years ago, we find the Lower Ordovician, which includes the Tremodocian and Floan periods. This is followed by the Middle Ordovician, from 471 to 460 million years ago, with the Dapingian and Darawilian. The Upper Ordovician completes this period from 460 to 444 million years ago. The latter is made up of 3 stages: the Sandbion, the Cation, and finally, the Hernantion. But why, and above all, how can we divide this distant period into several parts? During the 41 million years that make up the Ordovician, certain species appeared at very specific times. It is their successive discoveries, and in particular their dating, that have made it possible to establish the chronology of these 7 stages. The oldest, the Tremodocian, is defined by the arrival of the conodont species, Leptognathus fluctivagus, now extinct. This specimen is distinguished from contemporary conodonts by the lateral orientation of its main tooth-like projections. Traces of this fossil have been found mainly on the American continent, in Kazakhstan, and in China's Hebei province. The second stage, the Floan, marks the appearance of the graptolite Tetragraptus approximatus, an easily recognized H or X-shaped filter feeder. Traces of this specimen can be found almost everywhere in the world, with the exception of the UK, Africa, and Spitsbergen, an island north of Norway. The Dapingian stage, which heralds the beginning of the Middle Ordovician, corresponds to the first date of appearance of the conodont species Baltoniatus triangularis. The next stage, the Darawilian, is marked by the appearance of the graptolite Undulagraptus ostradentatus. The Huanghuachang section near the city of Yichang, China, is recognized as the world's reference stratotype point for these 2 stages. Finally, the Upper Ordovician saw the successive emergence of the graptolite species Nemagraptus gracilis, Diplocanthograptus caudatus, and Normalograptus extraordinarius in the Sandbian, Cattian, and Hernantian stages. Their stratotypic reference points are in Sweden, Oklahoma, the USA, and China's Hubei province. The Ordovician has left its mark all over the globe. It allows us to establish how the continents were laid out at this time in the planet's history. Geologically speaking, the Earth changed little between the Cambrian and Ordovician periods. Land masses are still mainly located in the southern hemisphere. The northern hemisphere is still almost entirely covered by the waters of the Panthalassa Ocean. Only the continent Siberia and a small part of Laurentia have found their way into this hemisphere. The supercontinent Gondwana, partly covered by shallow seas, occupies almost the entire southern hemisphere. It includes South America, Africa, India, Australia, and Antarctica. In the intertropical zone, the three small continents that have recently formed, Laurentia, which corresponds to North America, Siberia, and Baltica, lie in the tropics. Numerous volcanic arcs are scattered around, mainly bordering the coasts of the Iapetus Ocean and a newly formed ocean, the Reich Ocean. The opening of the latter further south between the Baltic and Gondwana initiated the closure of the Iapetus Ocean, but also the displacement of the supercontinent, which gradually moved away from the equatorial latitudes towards the South Pole. During this period, orogenic activity is very low. The mountain-building process was therefore limited to a few collisions between island arcs or ribbons of microcontinents against the south coast of the Laurentia continent. At most, a few small terrains or pieces of the Earth's crust, which detached from this megacontinent during the Lower Ordovician, will become attached to it during the Middle Ordovician. This geological phenomenon is known as the Taconian orogeny. It corresponds to a folding phase that lasted almost 10 million years before giving rise, after a long geological process, to the assembly line known today as the Appalachians. The separation of the continents reached its climax during the Middle Ordovician, the period of the Paleozoic era with the greatest number of continents. It was only much later, at the end of the Paleozoic, that they began to come together again. The Reich Ocean ridge showed strong activity at the beginning of the Ordovician. This led to the overflow of oceans into continental surfaces. It was during the Middle Ordovician that sea levels peaked, reaching, according to some estimates, 200 meters or 650 feet above today's values. While the tropical zone is the widest of known fossil times, by the end of this period, the supercontinent Gondwana had taken root at the South Pole, where it was gradually covered by an imposing ice cap. By studying the many fossils discovered in different parts of the globe, scientists are able to conclude that major climatic variations occurred during the Ordovician period. A considerable rise in sea level, as well as the proliferation of life in marine ecosystems over a given time, corresponding to the Middle Ordovician, demonstrate that drastic climate changes took place during this period. Similarly, the sharp decline or even extinction of many species suggests that some of these changes were probably rapid, leaving little or no opportunity for existing organisms to evolve serenely. During the Ordovician period, the climate was warm and tropical, especially during the lower Ordovician, when temperatures were at their highest. Some parts of the globe experienced record temperatures close to 60 degrees Celsius or 140 degrees Fahrenheit. However, towards the end of the period, around 445 million years ago, these temperatures began to fall, leading to a major ice age. The main continent, Gondwana, was gradually covered by a massive ice cap. This glaciation, which lasted approximately 1.5 million years, triggered the disappearance of a large number of animal species that were unable to adapt to the new environmental conditions. Thus, the Ordovician is a geological period characterized by the concomitance of a major ice age and one of the 5 greatest mass extinctions in the history of our planet. Indeed, despite tropical ocean temperatures that seem to exceed 30 degrees Celsius or 86 degrees Fahrenheit, the Ordovician left numerous indirect clues to the presence of ice on the continents during this same period. In an attempt to understand the mechanisms inherent in this climate change, scientists have turned to a computerized tool. They used numerical modeling that brought together several disciplines, namely meteorology, oceanography, and environmental physics. In this way, they succeeded in resolving the climatic paradox of this period. Comparison of the numerical simulations with a wealth of sedimentological, geochemical, and micropaleontological data suggests that the first stage in the growth of an ice cap occurred as early as the Middle Ordovician, more precisely during the Darwinian stage, as evidenced by the variations in sea level, attested for the same period. These results indicate that the presence of a polar ice cap is not incompatible with high tropical ocean surface temperatures. Gradual ice growth would then have allowed the ice cap to reach tropical latitudes, marking the onset of the Hernantian glacial maximum. In short, the Ordovician glaciation would have been a long-lasting one, of which the Hernantian, the last stage of the Ordovician, would in fact have been only the apogee. Thanks to a relatively warm climate, the Ordovician was a very prosperous period. Life already existing, but mainly located near the seabed, multiplied in an exceptional way. This period is defined as the event of the Great Ordovician biodiversification. During this relatively short period of only 25 million years, the oceans were truly filled with numerous families and genera of living organisms. There are many reasons for this unprecedented expansion of marine life. However, a key factor has been the increase in the surface area of continental shelves, particularly in the tropics, as well as high sea levels, all of which have encouraged the growth of phytoplankton, and consequently, of all aquatic organisms. These microorganisms have proliferated in the epicontinental seas, which are particularly extensive. Thanks to the abundance of phytoplankton, several groups of zooplankton made their appearance before expanding exponentially. These include chitanozoans and graptolites. In addition, constant volcanic and tectonic activity during the Ordovician period most likely provided the nutrients required for the development of planktonic life in the oceans. As a result, the water column gradually filled with all kinds of living microorganisms, but not only. Indeed, the first fish made their appearance at this time, and cephalopods, the ancestors of octopus and cuttlefish, diversified like never before, to the point where, by the end of the Ordovician, ecosystems comparable to those of today already existed. This period marks the biodiversity plateau of the Paleozoic. Life became so abundant in the oceans that it eventually began to spread on land too. Fossils of spores, the microorganisms that form part of the life cycle of many bacteria, plants, algae, fungi, and even some protozoa, have been found in sediments attributed to the Upper Ordovician period. Similar to contemporary liverworts, these first land plants were most likely descended from green algae, already widespread during the Cambrian, the preceding period. Their proliferation was confined to shorelines and coastlines, where they formed a low-growing carpet. Among the fossils studied from the same period, others revealed the presence of the first terrestrial fungi. They certainly played a major role in the colonization of emergent soils by flora, making mineral nutrients available to plant cells through mycorrhizal symbiosis. Although these discrete plants are fairly primitive and basic, they mark a decisive turning point in the evolution of life on Earth. And while this stage is still in its infancy, it is no less important. It's the first time on the planet, after bacteria and other microorganisms, that a form of life is actually visible on unsubmerged soil. Thus, during the Ordovician period, the hues that brightened up the ocean floor began to timidly color dry land. Although this transitional period is decisive in our planet's geological history, it has long been overlooked. Indeed, until the early 2000s, only two major lagerstätten, i.e. sedimentary deposits containing a wide variety of fossils, testifying to the Ordovician period, had been recorded. The first is located in New York State, USA. This is Beecher's trilobite bed, located in the Frankfurt Shale at Cleveland's Glen. It is associated with very particular environments, namely deep hydrothermal springs, in which low-diversity faunas dominated by a few arthropods thrived. Only 3 to 4 centimeters or 2 inches thick, this lagerstätten revealed numerous trilobites in an exceptional state of preservation, particularly of their ventral anatomy and soft tissues, the latter preserved by pyrite replacement. This mineral, which is an iron sulfide, allows X-rays to be used to study the finest details of soft tissue anatomy. The fauna of this site is largely composed of the trilobite, Triarthris atoni. It alone accounts for 85% of the organisms studied. Other species include graptolites, colonial filter feeders, branched algae, brachiopods, and other enigmatic taxa. Among the many trilobite specimens of Cryptolithus, Bellulus, Cornoproteus beecheri, and Primaspis crocetus have also been inventoried. In terms of population, it belongs to the penultimate stage of the Upper Ordovician, the Cattian. The second proven lagerstätten from the Ordovician to the 2000s was discovered in South Africa. This is the Soom Shale. It reflects the population of the Hernantian, the very last period of the Ordovician. Only the remains of pelagic organisms, i.e., those living in the water column, have been discovered here, thanks to their good state of preservation. These include arthropods, cephalopods, conodonts, and agnathan fish. However, these two conservation sites, like others, are far from providing as much information as those collected for the previous period, the Cambrian. All the more so as they provide new elements that only concern the end of the Ordovician. Nevertheless, as fossils are discovered, they continue to advance our understanding of this period. At the very beginning of the 21st century, in a valley in southern Morocco, north of Zagora, a major paleontological discovery revolutionized scientists' understanding of the earliest animal diversifications of the early Phanerozoic. This is the lagerstätten Les Phasota. The importance of the treasures delivered in this area even gives it the honor of being named by some the Ordovician Burgess Shale of Morocco. This storage site is an exceptionally well-preserved Ordovician deposit, but more importantly, it has yielded abundant and, last but not least, highly diversified marine faunas, some of which are attributed to the lower Ordovician. It has been selected as one of the 100 most important geological sites in the world because of its role in understanding Ordovician evolution. What makes the Phasota site so remarkable is its predominantly clay formation, dating from the Tremodocian, the very first stage of the Ordovician. This clay deposit, estimated to have lasted almost 2 million years, contains a wide variety of fossils, making it possible to study the paleofauna. Fossils of carapace animals, similar to those found in the Burgess Shale dating from the Cambrian, have been discovered in the Phasota clays. These have come as a surprise to paleontologists, who previously believed that this fauna had disappeared during the Middle Cambrian, some 40 million years before the Phasota clays were deposited. This paleofauna also reveals crown group taxa, whose presence was unknown in the lower Ordovician. While the Phasota fauna is dominated by trilobites, it also contains a high abundance of xiphosaurid arthropods, paleoscolicid worms, annelids, mollusks, brachiopods, and connular worms. Graptolites are also abundant. More recently, excavations at Taeshout, some 80 kilometers or 50 miles from the Phasota site, have uncovered a new fossiliferous site. It differs from the Phasota site in several respects. Firstly, it is a few million years younger. But above all, this site, which was once underwater, has preserved traces of large arthropods, described as giants. These, which may be related to modern creatures such as shrimps, insects, or spiders, must have been able to swim freely and move in currents. These organisms are in the minority in the Phasota area. Given their colossal size, we imagine that they almost certainly dominated the seas 470 million years ago. Some specimens could measure up to 2 meters in length. They are therefore much larger than any specimens discovered to date. These giant arthropods, although not yet fully identified, may belong to species already discovered on the Fuzuta coast. Their large size and free-swimming lifestyle suggest that they must have played a unique role in Ordovician ecosystems. If paleontologists can study mineral elements, they can also observe with precision the soft parts, which are in an exceptional state of preservation. At last, scientists are able to study the anatomy of the first animals the Earth ever sheltered. The carcasses discovered are thought to have come from a relatively deep marine environment, which is completely different from previous discoveries, where carcasses were preserved in shallow places. In addition, animals such as brachiopods were found attached to some fragments of these giant arthropods. This suggests that these large animals, once dead, served as nutrient reserves for seabed communities. The remains of Teichut's fossilized organs enable scientists to map the marine environment of the early Ordovician period with some precision. The Ordovician remains a largely unknown period, yet it was a pivotal moment for the evolution and diversification of organisms that appeared earlier in the Cambrian period. While data concerning this period was relatively scarce, the discovery in 2020 of a new site, extremely rich in fossils, whose quality of preservation rivals the most renowned Cambrian sites, may partially fill the remaining gaps. This is the Castle Bank site, located in England in central Wales. Its sedimentary deposits and fossils date back to the Middle Ordovician, around 462 million years ago. More than 170 different species have been recorded, the vast majority of which are completely new to paleontologists. Unlike the Fuzuta site in Morocco, the Castle Bank site contains a veritable miniaturized ecosystem. Indeed, most of the fossils are very small, measuring no more than 1 to 3 millimeters in length. They attest to a significant reduction in size compared with the previous period. However, their state of preservation is so exceptional that they provide researchers with a wealth of new information. Among the species studied, crustaceans, crabs, worms, sponges, and many others have left fossilized traces of their internal organs, such as their digestive tract, and even their nervous system. Soft tissues such as eyes, optic nerves, and even brains can be exploited. Likewise, the tiny, fragile limbs of arthropods are preserved, as are their sophisticated fine tentacles for filtering seawater. Among the specimens uncovered, some unusual groups of animals are brought to light. Opabenids, arthropods with a vacuum cleaner like proboscis, are described, as are wuaxids, oval-shaped mollusks with soft bellies and backs covered with rows of scales. Other insect-like creatures probably distantly related to this category have also been described. Now let's delve into the depths of this period to take a closer look at these strange creatures. While the planet enjoyed a warm, humid climate, if the land was still bare and virtually barren of life except on a few shores, the shallow seas were teeming with life. Brachiopods were extremely abundant during this phase of evolution, still largely dominated by invertebrates. Brachiopods are animals with shells whose soft parts are contained between two valves. Although they resemble bivalves like clams, the body parts inside the shell are very different. What's more, their valves are symmetrically different from those of clams. A pedicle emerges from the larger of the two valves, enabling this brachiopod to attach itself to the seabed. Brachiopods are protostomans, one of two existing groups of bilaterians, along with bryozoans. Bryozoans are mossy animals that live in colonies. They take a variety of forms, including arborescence or lace fan. Although they strangely resemble corals, they are not corals. Each individual secretes an arborescent calcium carbonate skeleton attached to a hard surface. Each specimen, called a zooid, usually measures less than 1 millimeter in length and lives in its own little lodge. All interconnected, they form colonies containing millions of zooids, which can grow to several meters in size. Within the same colony, each individual plays a distinct role. Some gather food, others help to strengthen the group. Finally, some of these individuals contribute to cleaning. To feed, bryozoans create a water current that brings tiny creatures to their mouths. Most of the time, these are plant-like organisms such as phytoplankton. Bryozoans gradually colonize certain areas of the seabed, eventually building up into extensive reefs. Trilobites make up a large part of the Ordovician fauna. These crustaceans, which secreted an easily identifiable segmented exoskeleton, reached their peak during the Ordovician. In fact, they are the most famous prehistoric animals after the dinosaurs. Over 18,000 species have been recorded. They ranged in size from 1 to 70 centimeters or 0.3 to 30 inches in length. And their way of life covered the whole range of thalassic environments. Some were detritus feeders, grazers, or even predators. While a few species mastered swimming, the majority of trilobites moved along the seabed, burrowing into the sediment to escape predators. Trilobites also developed the first advanced visual system. All trilobites had a head, called the cephalon, a thorax and a tail, as well as pairs of biramid legs on the ventral side. As with crustaceans, these legs had two branches, one serving as a gill for respiration, the other for locomotion and feeding. Among the most common trilobites, Cryptolithus measured around 1.5 centimeters or 0.6 inches in length, and Acidaspis nearly 2 centimeters or 0.8 inches. But there were also specimens of far more spectacular dimensions. These included the calamine, between 2 and 5 centimeters or up to 2 inches. And above all, the Isoletus, which could commonly reach 38 centimeters or 15 inches in length. However, the world's largest trilobite species ever discovered is Isoletus rex. Its complete fossil measures no less than 72 centimeters or 28 inches long, and 40 centimeters or 16 inches wide. The seabed was also covered by a large population of arthropods. Eurypterids, or gigantostracans, are one of the many species. They are also known as sea scorpions, for their shape is similar to that of modern-day scorpions. Eurypterids have elongated bodies extended by a long articulated tail. Its thorax is equipped with fearsome pincers, and its head features several pairs of eyes. A pair of legs that were intended to act as fins is terminated by large swimming paddles. Finally, most species are equipped with a tapering string at the end of the tail, which may well have served, if not to inject venom into some prey, to lacerate it. The size of eurypterids was highly variable and probably depended on environmental factors, lifestyle, and taxonomic affinities. While the majority of arthropods were small-bodied and probably scavengers, the eurypterid was undoubtedly one of the most feared predators of the Paleolithic oceans, commonly measuring a meter in length. Some of these specimens grew to over 2 meters or 7 feet in length. Megalograptus is another arthropod species. It is the oldest known eurypterid. Of good size, the largest of the specimens discovered measures 78 centimeters or 31 inches in length. Like its congeners, it has spikes on its third pair of legs. Large spines on its various appendages helped it to locate prey, hidden in soft sediments and out of sight. Its telson, the last segment of its tail, if not venomous, probably helped it to tear its victims apart when feeding. During the Ordovician period, many new forms of echinoderms appeared, including sea stars, sea urchins, and crinoids. At that time, there were some 20 classes. While all present-day echinoderms have a pentarad configuration, i.e. 5 rays, this wasn't always the case for primitive forms, such as the carpoid, a strange specimen that still holds many secrets. Its truncated tail turns out to be a single arm at one end of its body. The animal's mouth is concealed on its underside. This single arm was probably as much for moving around on the seabed as for feeding. Given its morphology, it was probably a detritivore. The Ordovician period is also marked by the presence of large numbers of graptolites. These animals, living in colonies of 3 to several thousand individuals, belong to the Hemichordae phylum. They resemble coral polyps, made up of coiled toothed tubes, and are gathered around a main stem, on which small cells shelter each individual specimen. These colonies of animals, floating or attached as the case may be, measure from 10 millimeters to almost a meter in length. The names of the different graptolite genera refer to the appearance of their branches and their number. Monograptus, for example, has a single linear branch, while Didymograptus and Tetragrammatus have 2 and 4 respectively. Spirograptus has a single spiral branch. Cephalopods are the dominant predators. Cephalopods lived in front of a long shell, called a chambered shell, because it was made up of several partition cavities. While some, like ammonites, had spiral shells, others were classified as orthoconic cephalopods. As their name suggests, they had a straight shell. This could be extremely large, reaching up to 6 meters or 20 feet in length, in the case of Camarasaurus. Another species, Treptoceras, was usually around a meter long. Cephalopods were formidable predators, moving at will through the water column. It controlled its buoyancy by varying the amount of air in the various chambers of its shell. It was able to move by reaction, forcefully expelling the air contained in its body. Like a squid with a protective shell, this predator was at the top of the food chain. Thanks to its strong tentacles, it could dexterously capture any coveted prey, especially the trilobites that lived in great numbers on the sediments. All he had to do was carry his trophies to his mouth. This beak-shaped mouth was located in the center of the tentacles. In modern cephalopod groups, this shell has since shrunk considerably, or even disappeared altogether. Today, for those specimens that still have it, it is no more than a relatively small internal structure. Modern cephalopods include squid, octopus, nautilus, and cuttlefish. To feed all these little creatures, numerous mollusks line the seabed. Among them is the group of pelecypods, or bivalves. This group includes clams, scallops, oysters, and mussels. These filter feeders, which form shellfish beds, have two identically shaped valves on each of their body, and a laterally compressed burrowing foot. The upper edge of each shell usually has several teeth and sockets that fit into those of the opposite shell to form a hinge. Other mollusks also lived in large numbers on the seabed, gastropods. Snails and slugs also played an important role in this biodiversity. Unlike snails, slugs do not secrete shells, but they do have the same internal organs: a heart, a digestive system, gills, and a nervous system. Snails can be recognized by their unique coiled or spiral outer shell. The shell offered refuge from threats, which must have been incessant. The Ordovician seabed was also littered with corals, each more colorful than the last. Some were horn-shaped and secreted an exoskeleton that could grow up to 1 meter or 3 feet long, such as the Rugosa coral. Although this coral could be solitary, it could also form large colonies by grouping together with many of its companions. Like all corals, these are much more than just a stony skeleton. Inside, animals in the form of seeded polyps are evolving, as is their framework, which adapts to their growth. Equipped with tentacles containing stinging cells, they were able to capture their prey in suspension. While most corals captured and devoured their prey, they drew most of their energy from algae. Numerous varieties of round tabulate corals also carpeted the ocean floor, covered by warm, shallow waters. But the most extraordinary species is undoubtedly the crinoids. These marine animals gave their environment the appearance of a spring meadow. Indeed, they are commonly referred to as sea lilies. Belonging to the phylum Echinodermata, which also includes sea stars, sea cucumbers, and sea urchins, they display the pentarad symmetry typical of echinoderms. Attached to the seabed by a long pedicle that can reach up to several dozen meters, they look more like flowers. Yet they were actually animals that fed with the help of podia, hollow cylindrical organs on their delicate arms that resembled the lightness of feathers. Food particles suspended in the ocean currents were captured by these arms before being placed in a furrow running through them. Then, like pistols, tiny cilia carried the trapped particles to the mouth at the center of the head. Among the invertebrates inhabiting the Paleolithic seabed, there are also those belonging to the sponge group. They played an extremely important role during the Ordovician period as reef-building organisms. Stromatoporoids, are sponges calcified in layers called lamellae. Between each of these plates are vertical structures called pillars, as well as small distinct chambers called galleries. These galleries are lined with choanocytes, which serve to capture food and breathe while water circulates between the layers. Bumps are visible on the surface. These are nipples that probably contributed to the absorption and flow of water through this architecture. As fossils testify, stromatoporoids coexisted during the Ordovician period with many other species such as mollusks and brachiopods. Some shell organisms may even have been completely enveloped by a growing stromatoporoid, with some reaching 30 centimeters or 12 inches in diameter. However, these two species cohabited in harmony, with water flowing from one end of the stromatoporoid's porous structure to the other. Another sponge, with a very distinctive shape, also dominates the seabed. This is Brachiospongia digitata, a glass sponge. This siliceous sponge has a skeleton made up of a network of opaline siliceous spicules. Its cup-shaped structure features a series of 6 to 12 curved finger-like extensions. Finally, there's the Aulacera plumeri. This elongated organism resembles a trunk. Some of its specimens have probably reached 5 meters in height. We can imagine these animals living in colonies, giving the seabed the appearance of a more or less dense winter forest, through which prey and predators had to thread their way or even chase each other. Finally, the first vertebrates visible during the Ordovician period were agnathans. These fish are still the first vertebrates to appear on the planet. They are organisms that lack jaws and mandibles. Their mouths were slit-shaped, round as in the modern lamprey and hagfish, the only survivors from this period. Most agnathans had a cartilage skeleton and at least 7 gill pouches. These fish lacked paired fins and a stomach, but had a pineal eye, which was particularly sensitive to light. Agnathans could measure up to 30 centimeters in length. The plant world also underwent a number of diversifications during the Ordovician period. Encrusting, calcareous algae, green algae, and brown algae multiplied and diversified, to the detriment of cyanobacteria. Although cyanobacteria played an essential building role in reefs, they were gradually supplanted by corals and bryozoans. From the Middle Ordovician onwards, more precisely, during the Darwinian period that stretches from 467 to 458 million years ago, the fauna underwent great diversification. While no new phyla appeared, the number of families quadrupled, and the number of genera tripled. Pelagic animals were far more abundant than in the previous period, giving the seas the appearance they would retain throughout the Paleozoic. This vast diversification is probably explained by the drop in ocean temperatures, which were quite high at the beginning of the Ordovician, exceeding 45 degrees Celsius at the time. Indeed, temperatures gradually decreased until the Darwinian, when seas were comparable to today's equatorial seas, with waters around 30 degrees Celsius. Conditions for the development of life had then become particularly favorable, hence this great diversification. In addition, oceanic waters were enriched in mineral salts by the intense activity of ridges, and, by continental erosion, accentuated by the growth of terrestrial plants, especially as a large part of the continents was covered by shallow waters. These conditions favored the development of phytoplankton and algae attached to the ocean floor. As a result, food chains were literally boosted, encouraging the diversification of filter feeders, grazers, and primary consumers of all kinds, and then, de facto, that of predators. By the end of the Ordovician period, a quarter of all marine animal families and half of all marine animal genera had disappeared. This was the first great extinction of life, better known as the Ordovician crisis. But what could have happened on the planet to bring it to this point? According to scientists, the disappearance of more than 70% of all species is due to an immense episode of glaciation that the Earth underwent. According to some researchers, this biological crisis can be broken down into two extinction phases, spanning some 1 million years. A climatic cooling, triggered by continental drift, would have led to a drop in ocean temperature. Then, induced glaciation would have led to the formation of an ice cap, extending as far south as the high latitudes. Cold, oxygen-rich water would then have spread to areas previously poor in oxygen, causing the disappearance of organisms adapted to anoxic conditions. As ice expansion led to lower sea levels, oceans would have lost large quantities of water. As a result, organisms living in shallow waters would have suffered greatly. Then, at the end of this period of glaciation, rising sea levels would have triggered an expansion of the waters, this time warm and poorly oxygenated, leading to further extinctions. Another hypothesis challenges this explanation and suggests that this first of 5 Phanerozoic mass extinctions could be attributed to volcanic eruptions. Volcanoes are known to emit mercury. However, mercury enrichment of sediments has already been observed in other, more recent biological crises on the planet. A team of researchers therefore analyzed sediments located in China's Hubei province, where the Wengejiawan Cut, a Hernanchian stratotype, is found. During the Ordovician, this region belonged to the Yangtze Block, then covered by the sea. Black clays rich in graptolite fossils were deposited in an anoxic environment. Then, as the sea level dropped and the water cooled, clayey limestone covered these clays. The latter contains fossils of the Hernanchia fauna, characteristic of this ice age. The subsequent clays represent the post-glacial rise in sea level. However, it turns out that mercury enrichment was measured just before the clay limestone was deposited and persisted throughout the glaciation. A second enrichment occurred immediately before the arrival of the post-glacial clays. Similar observations were made in Nevada, when this territory belonged to an isolated continent, the Laurentia continent. Scientists have deduced that these mercury emissions correspond to peaks in volcanic activity. These two peaks of activity coincide with the two extinction phases of the Ordovician period. Researchers therefore hypothesize that eruptions attributed to supervolcanoes would have released gigantic quantities of carbon dioxide and methane, leading inexorably to a drop in Earth temperatures and the development of polar ice caps. For other paleontologists, the ice age that the Earth experienced around 460 million years ago could have been caused by a much more extraordinary phenomenon, one that is extraterrestrial in nature. In other words, winter came from the cosmos. A first hypothesis mentions a violent collision between two asteroids. The collision would have occurred in the asteroid belt between Mars and Jupiter, and its unprecedented violence would have shattered the larger of the two objects, which were over 150 kilometers or 93 miles wide, into billions of pieces. According to this hypothesis, the numerous fragments, some no larger than dust, others several tens of kilometers or miles in size, then began to orbit the Sun. The trajectory of the smaller debris changed and it gradually moved closer to our star, and consequently to Earth. 10,000 years after this extraordinary collision, the dust began to penetrate the Earth's atmosphere and scatter all around our planet. The result was a shower of fine particles that lasted for almost 2 million years. But after these tiny pieces of debris, larger and larger rocks, similarly deviated from their orbits, impacted the Earth. By analyzing numerous asteroid fragments in Ordovician rocks, scientists were able to date this event back 466 million years. In fact, the quantity of extraterrestrial dust is 1,000 to 10,000 times greater than in the sediment layers below this period. If debris from outer space regularly falls to Earth, the colossal quantities that reached it at that time cannot have been without consequence, and with good reason. Through this curtain of dust, the sun's rays had difficulty reaching the planet. Temperatures dropped considerably. Snow appeared at the South Pole and fell without interruption. As it accumulates, it gradually turns to ice. The South Pole is gradually covered by an ice cap, trapping a significant quantity of our planet's water. As a result, ocean levels dropped and winter set in on the planet. This winter will continue for millions of years. However, not all scientists agree with this theory, for while it is undeniable that an ice cap did indeed form at the South Pole between 467 and 458 million years ago, or even earlier, temperatures had already begun to fall by the beginning of the Ordovician period. What's more, for many experts, a veil of dust from outer space could not have influenced climate over such a long period. Other mechanisms probably contributed to this drop in temperature. What if the continental waltz had something to do with it? During the Ordovician period, 3 small continents drifted away from the megacontinent Gondwana. While the latter remained at the height of the South Pole, the smaller continents moved closer to the equator. Laurentia, Siberia, and Baltica therefore enjoy a tropical climate. The sun's scorching rays alternate with unparalleled precipitation. The carbon dioxide in the atmosphere is then precipitated by rainwater onto the continents or directly into the oceans. Under the effect of this heat and abundant rainfall, rocks are dissolved. They are then eroded by the ions released by this dissolution. Some of these ions contain carbon, which is also carried by runoff into the oceans, where marine organisms use it to build their shells and skeletons. Carbon dioxide is inevitably reduced in the atmosphere, so it no longer fulfills its greenhouse function. As terrestrial temperatures depend directly on the amount of carbon dioxide in the atmosphere, If the latter falls, so do temperatures. So, for some scientists, the cooling of the planet during the Ordovician period is explained by plate tectonics. Finally, another theory, albeit controversial, could explain this mass extinction. This is linked to the supernova explosion of a dying star, several thousand light-years from Earth. This explosion, which seems to have taken place over 440 million years ago, in the farthest reaches of our galaxy, generated powerful radiation energy, releasing an extraordinary quantity of gamma rays. After a long journey at the speed of light, these unfortunately hit the blue planet after piercing the atmosphere with unprecedented power. The impact was so violent that it annihilated the air molecules and swept away the atmosphere surrounding our planet. One of the first consequences was a chemical change in the atmosphere. Nitrogen and oxygen molecules were transformed into nitrogen dioxide. This brownish, particularly noxious gas eventually surrounded the planet. The planet was then no longer able to receive sunlight, causing temperatures to plummet. But let's put this into context and look at the various phases of this extinction, which have been extensively documented in a 2010 TV documentary. Before this supernova, life teems in the oceans. The sun was shining brightly under a radiant sky, and species were multiplying on a massive scale in tropical seas. For the first time on the planet, which is warm and humid, life seems to be gaining ground. Some species are even beginning to venture out of the water, particularly in the shallow, epicontinental seas. It's true that on the surface, paradoxically, life is hardly possible, and our world looks more like a wasteland, like Venus, for example. Oxygen levels are so low that no modern animal could breathe properly. Imagine the silence that must have reigned on the planet at that time. Absolute silence. But this Olympian calm is about to be disturbed by the supernova explosion of a dying star 6,000 light-years from Earth. Powerful radiation bursts from its poles. It's a gamma-ray explosion, the most violent phenomenon in the cosmos. In just 10 seconds, this supernova produces more energy than 1,000 suns put together. These gamma rays then travel through space at the speed of light. Despite its remoteness, planet Earth will unfortunately not be spared. But luckily, the cataclysm occurred far enough away that it didn't blow away our atmosphere and send it into the cosmos. Gamma rays are therefore able to penetrate the upper layer of the atmosphere. Like the northern lights, they produce a spectacle of rare beauty, but their impact will be decisive and above all destructive. Within seconds of the supernova, these lethal rays annihilate air molecules and blow away the atmosphere. A star of strange behavior stands not far from our sun, visible even in broad daylight. These are the remnants of the supernova, which will remain visible for a few more months before disappearing completely. At first glance, this distant cataclysm seems to have had little impact on life on Earth, and yet, in the hours that followed, extinction did indeed begin, but at a microscopic level. During their passage, the gamma rays destroyed 30% of the ozone layer, allowing the sun's ultraviolet rays to reach the planet. The light is so bright that it appears 50 times more intense than in the middle of the Sahara Desert. If terrestrial creatures had existed at this time, they would literally have been consumed in a very short time. The consequences of this biological disaster will begin to show in the water. Plankton, invisible to the naked eye, are the first to suffer. The sun's rays, which have become too powerful, burn off the vast majority of these microorganisms. Plankton is the basis of the food chain. Imagine the panic of plankton eaters as they watch their food run out under the devastating sun. Trilobites, whose larvae's main incubation site is the water's surface, see their reproduction diminish and even come to a halt. Nothing can survive the intensity of the sun's rays. Many corals and other animals quickly perish. A veritable domino effect leads to the collapse of the entire food chain. Tropical zones are the first to be affected by this particularly harmful ultraviolet radiation, and coral reefs are gradually dying out. Soon this phenomenon spread to the four corners of the globe, with corals succumbing to starvation. More and more animal species run out of food, and the reefs die out week after week. In the months that follow, the weakest animals are the first to disappear. For some, driven by their survival instinct, eating habits change. Predators accustomed to choosing their food must now make do with what they are lucky enough to find. Some even start eating corpses, something they would never have done before. Some species will eventually leave the dying reefs in search of food. This is particularly true of the giant orthoceras, the squid's ancestors, who have preferred to venture into brackish waters to devour new prey, the eurypterids. Famine rages. It affected all species, causing more and more extinctions. Biodiversity underwent an unprecedented crisis. If all the creatures in existence at the time had disappeared, what would have become of our planet? Would life have evolved in a completely different way? More on this later. In the months that followed, the scattered molecules of the atmosphere swirled around the Earth's sphere. As the atoms line up, they form nitrogen dioxide, a brownish, noxious gas. The bright sunlight is gradually masked by this thick layer of putrid air. After suffering the sun's wrath, the planet now receives only 50% of the sun's rays. Inevitably, the temperature drops to -12 degrees Celsius or 10 degrees Fahrenheit. Species that have survived famine until now are now faced with temperatures that are far too low. Nautiloids in particular are seeing their eggs laid a few months earlier fail to develop properly. The cold has slowed embryonic growth, encouraging the appearance of bacteria. The rare specimens that survive this compromised incubation enter a world of chaos. During their short life, they have only one objective: to find food. But these mollusks have a distinct advantage over many other species. Their bodies are totally protected by a shell that has enabled them to live in deep waters, sheltering from the devastating effects of climatic cooling. What's more, they have a varied diet. They are sometimes even labeled as opportunists, and have never hesitated to be scavengers. Animals that were able to move around, therefore, tried to reach milder areas. Unfortunately, during the Ordovician period, the majority of animals were sedentary, and the seabed was largely occupied by corals, brachiopods, sponges, and many other non-mobile species. Another problem associated with the lack of food is shell formation. The shells of most reef dwellers are made of calcium carbonate. Calcium carbonate is extremely difficult to secrete in cold water, which has inevitably led to the disappearance of many species. The planet's cooling has also resulted in a harsh and, above all, unpredictable climate. Before the supernova, the weather was always very stable. But for the first time in millions of years, major temperature shifts are triggering strong ocean currents. These generated violent climatic disturbances. The planet, hitherto relatively peaceful, is confronted with a Category 5 hurricane that sweeps across its surface on a permanent basis. Many animals, too weak to resist, end up smashed against the rocks. Those still able to fight try to find refuge in the depths, away from the surface waters, which are far too turbulent for them. But not everyone can escape the pressure of deep water. This is also the case for orthoceras, which, despite their titanic size, cannot find shelter there. Their disproportionately long shells are not designed to withstand such pressure. As a result, even the largest predator known to Ordovician times found itself in an awkward position. Nautiloids, on the other hand, thanks to their more robust shells, were able to find refuge in deeper, less turbulent waters during these storms. For dozens, even hundreds of years, these species suffered heavy losses. Corpses by the thousands washed ashore. More than 500 years after this event, from outer space, almost a third of the fauna became extinct, and the remaining creatures had little chance of surviving in these conditions. Life on Earth, in whatever form, was threatened with complete extinction. However, a certain number of individuals would survive and make it through the following thousands of years. Nearly 150,000 years later, life on Earth was virtually reduced to nothing. The species that survive this biological catastrophe are constantly starving. Orthoceras, once lords of the seas, are now weakened scavengers in a world that no longer resembles what it once was, especially as a new ice age begins. The planet is getting colder and colder, and snow is piling up at the South Pole, until over the centuries it has become so dense as to form immense glaciers, several kilometers or miles thick. These run along the length of the Gondwanaland continent, spreading further and further north. These glaciers trap a huge quantity of water, so much so that sea levels drop considerably, losing up to 60 meters or 200 feet. As a result, the shoals dry up completely, squeezing the life out of the few remaining communities. This drying up proves even more fatal than the gamma-ray explosion itself. Some animals showed great resilience during this new ice age, in particular the astrapus, the ancestor of all vertebrates. The first known vertebrate fish, this jawless agnathan, was able to follow the retreating ocean. Measuring around 10 centimeters or 4 inches in length, and equipped with large external bony plates, serving as a shield, it withstood cold water better than any other aquatic creature. What's more, since its diet was not exclusive, it fed on whatever it could find. Carnivores, on the other hand, which fed strictly on a specific prey, found it much more difficult to feed, and many of these species became extinct. What's more, Astrapus didn't just adapt to its environment. In fact, a new variety of fish soon appeared. These now have bones and muscles that give rise to the first gills, making it easier to sort and absorb food. In this totally transformed world, Astrappus was not the only fish to follow the movement of the oceans. The Eurypterid, commonly known as the sea scorpion, also changed its territory, fleeing from the surface. It has even taken the place of the Orthoceras in the food chain, its main rival, which did not survive these extreme conditions. Since Astrappus was half its size, its need for food was much lower, a much more favorable factor for its survival. During this ice age, the planet took on a completely different aspect. Falling sea levels reveal new landscapes. The reefs created by sponges, corals, and arthropods were reduced to skeletons and partly emerged. The rare species that survived these reefs did not survive the cooling, especially as glaciers gradually invaded the subtropics. Gigantic icebergs drift through these former tropical regions, crushing everything in their path, including coral, thousands of years old. Year after year, sea levels continue to fall. Life is hanging on by a thread, but those still alive are struggling to keep up with the oceans, while aquatic habitats are becoming ever smaller. Unfortunately, the cascading consequences of the supernova are not yet over. Soon, the survivors will have to face a new climatic event, as more than 500,000 years after the start of this crisis, the planet is showing signs of warming. Glaciers are beginning to melt. Species that managed to adapt to this ice age will once again have to acclimatize to these new temperatures to stay alive. Some species stand out by adopting unprecedented behaviors. The Eurypterid is the best example. This sea scorpion, now the world's largest predator, began carrying the carcasses of its prey towards the shore. It even ventured onto land, as evidenced by the development of lungs found on certain specimens. Astrappus's cousins, the acanthodians, have also developed jaws. They are no longer content to eat algae. These scavengers can now cut flesh and feed more easily. In this way, they moved up the food chain. In the course of this long adaptation, the roles were reversed. Predators became prey and vice versa. But no matter, life, fragile as it may seem, has endured. More than 3 million years after this extinction, it's time to take stock. And it's a heavy one. Over 70% of species have become extinct. According to scientists, nearly 1 million species existed before this mass extinction. This means that around 700,000 of them died out in this relatively short period on a global scale. All living organisms were impacted, bottom dwellers and pelagics alike. Yet its impact on the history of the living world was not so catastrophic, insofar as this biological crisis did not result in the total disappearance of any major animal group. Life proved to be extraordinarily resilient, beyond all expectations. At the end of this period, the Earth will gradually return to a tropical climate, giving the various animal and plant species a little respite. But will this respite be long enough to allow new biodiversification? I think you already know the answer.